Okay, good morning, everybody. I think it's time to go ahead and get started. We still have some people getting some food and back and getting checked in, but we've got a full agenda, so we should probably go ahead and get started. My name is Rodney Sutter. I'm a program director of therapeutic studies at the SWOG Data Operations Center in Seattle. And I'd like to welcome you all to, I'd like to say our first in-person training course. We did have one last year in Seattle, but it was not as full. We kept people spaced out a little bit more. So it's kind of exciting to have a full room back after, I think it was 3 or 4 years ago, we were in San Francisco. In addition to this, we're carrying on what we've done for the past number of meetings is having the ability for remote participants to watch remotely. So I think we ended up with about 120 registering for the course in person and about 150 online. So that's a pretty good turnout. So before we get started with our presentations, I just want to take a few minutes to go through our agenda, talk about the goals of the training course, and cover the CRA workbench and the ORP manual a little bit. So the goals of this training course is to introduce the fundamentals and expectations of SWOG and NCI policies and procedures for new CRAs. I highlight new here because we understand a lot of you have experience elsewhere before SWOG, so it's just new to SWOG. To illustrate the process for protocol development, data submission, specimen submission, SAE reporting, and audits, and then also provide the foundation to efficiently perform your responsibilities as a new SWOG CRA. So here's a copy of our agenda. Like I said, it's pretty full. We do have 2 breaks planned here in the middle, and then we'll break for lunch and then come back into a different meeting room for the practicum session this afternoon. As a reminder to our remote participants, the afternoon session is just for the in-person attendees. And if you'd like to complete the practicum, you can do so online by going to the SWOG website and click on training. So this training course is provided free of charge thanks to the representation on this slide. The Hope Foundation provides a generous grant that covers the meeting room, the continental breakfast, the manual, and other supplies that we have. And then CRAB and SWOG contribute to the data management activities that go into planning this course, which we start months prior to this. I mentioned the ORP manual. It's a daily reference tool for CRAs and RNs. It's available on the CRA workbench, and it really should be used for your day-to-day activities in addition to what you learn in today's training course. Has administrative and data management resources, response assessment criteria, forms completion guidelines, among a number of different other sections. So can I get a quick show of hands? How many have been to the CRA workbench? That's a good number. I typically ask this at each training course and in other forums just because we do mentoring workshops where we go out to different sites. Our data coordinators answer questions by email and phone, and oftentimes we refer to the CRA workbench and are surprised how many don't even know that it exists. So I'm not going to go through all of the links that are here for you. I'm just going to highlight a few. I already showed you right there, the ORP manual. If you click on tools, the tools folder, you'll have things like the BSA calculator, clinical trial review guide, creatinine clearance calculator, date counter, ideal body weight calculator, and a QT interval. Under resources, you'll find some other important links. Best practices for SWOG studies. Highly recommend you check that out if you're not aware that it's there. It'll give you guidance for things that aren't clearly outlined in a protocol. We have the CRA newsletter archive, CTCAE, data submission guidance and tips, and then frequently asked questions, the SWOG glossary, specifics about the ORP committee within SWOG, and SWOG policies. If you click on the patient reports and data quality folder, that's where you'll find the DQP for RAISE studies, your expectation reports, find out which patients at your site have been made ineligible. Your IPR reports will be there, any patients you currently have in follow-up, and any outstanding queries in a query report. And then under study reports, you'll see accrual by site, race, sex, disease site, BMT and RT facilities, SAEs for a given study, studies in long-term follow-up, and studies with that no longer require follow-up. And then down below here, there's a contact us link. Oftentimes, you're not sure who to address your question to. So we have a number of distribution lists that are committee specific. These reach all the data coordinators that are assigned to that committee. So even if your data coordinator for the study that you're asking about isn't available, we'll have backup data coordinators that can get back to you in a good turnaround time. In addition, there's one specific for lung map. And then we also have email distribution list for questions about your expectation report. If you need to contact CTSU, if there's something technical related, something that's not working technically within the website that you need to ask a question on. And then if you're just really not sure overall, we have a data management link that will reach myself and the supervisors in our group. I encourage you guys to join the CRA mailing list. We don't overuse this and fill up your inbox with a bunch of things, but we do notify you if there are office closures, if there's changes to policy or something that's more of a major change. We'll use that to send notification out to you. And then as far as the CTCC evaluation form, for the first time, we used to always collect these on like paper copies you'd fill out at the end of the course. We're going to do all of this online this time. So at the end of the course, you should receive an email from one of our CTCC coordinators and sending you a link to the evaluation form. Try to keep some notes as you go along. If you have any feedback for us, for the presenters, the content, the material, ideas for a future CTGC, we'll keep that in mind. And then just fill it out online at the end of the course. You will need to complete this to get your certificate of completion for the course. And with that, I'm going to go ahead and turn things over to Dana Sparks, who is our Director of Operations and Protocols at the SWOG Operations Office in San Antonio.
Good morning and welcome everyone to SWOG and to San Francisco. Hope it's going to be a great group meeting. Some of you know we are having our election for our next group chair at this meeting, so it'll be an eventful meeting. There's a lot of opportunities during the meeting to learn a lot and to network, so I hope you'll be able to take advantage of that. So I have the privilege and responsibility to talk to you a little bit about protocol development in SWOG. So we'll be talking about clinical trials, the way they're designed, how we develop protocols in SWOG, and some of the details about protocol documents. So preclinical background to start with, the different types of clinical trials, protocol development, key protocol sections, and protocol actions. So those are the things that can take place on a protocol after it's activated. So to talk a little bit about preclinical background, you probably are aware of this, but to give everyone a baseline understanding, there's a lot of work that goes into the development of a new trial before it ever gets to use in humans. So whether we're talking about a brand new drug or a combination of drugs or a combination of a drug with a radiation regimen, there's a lot of work that happens preclinically, and that includes often development of a molecule up front. And this can come into being in a lot of different ways, including anecdotal evidence from natural products, or design of a molecule that targets certain proteins on a cancer cell. And of course, with the newer drugs, trying to create ways to make a person's immune system react to the cancer. So there's often many years of study that happens long before a new drug comes into clinical trials. All of that information gets filed with the FDA before, usually a drug company can get approval for introduction of this new compound into humans. And usually the last step before all of that information is filed with the FDA is testing in animal models. And so a new compound goes through a lot of different stages and steps before it can ever be used in human beings. And then when it's introduced into humans in clinical trials, there's a stepwise process for that as well. And the different types of clinical trials, we'll kind of walk through them. A phase 1 trial is usually designed to determine the maximum tolerated dose. Now that's the classical design for a phase 1 trial, but maximum tolerated dose is kind of shorthand for a lot of different considerations, including how best to give the drug, if that's IV versus oral, or is this compound better absorbed if it's given with food or without food? And do you decide to give it once a month, or is it a daily administration? So there's a lot of work that goes into figuring out what's the best way to give this drug. And then determining the maximum tolerated dose is a model many of you are probably familiar with, where patients are registered in stages, and they're registered at lower doses proceeding to higher doses to see what the side effects of the drug are. Usually phase 1 studies are very small. You're generally talking about maybe 20 patients or 30 patients at the most. They tend to be very focused on safety and analysis of any adverse events. And the goal of those studies is both to develop anecdotal efficacy information to try to figure out, is this drug really working, but primarily to make sure that it can be given in a safe way. If a phase 1 study is successful and a dose is established for use in future studies, then a phase 2 study might be done. And these studies are really designed to narrow in the efficacy question and to take a look at patients who are very similar to each other. So that's why you see a lot of times in phase 2 studies, you have very specific and narrow eligibility criteria. The intent is to make the patients as much alike as possible so that then you can really look at what are the effects of the drug. Now, what you will often see in phase 2 studies is that there may be underlying genetic components that we're not aware of, and that drugs may be effective in some patients and not in others because of those. So often phase 2 studies don't get you the real information that you need to have. But they do tend to establish that a drug can be effective in particular groups of patients. Usually phase 2 studies are designed around a tumor response endpoint, not always, but that's usually the practice, and they tend to be kind of medium-sized. So phase 2 studies can be single-arm phase 2s, and those usually are about 25 to 30 to 40 patients, or you can have a randomized phase 2 that is to kind of establish not a direct comparison with the standard of care, but at least to have a contemporaneous group of control patients so that you can make sure that you're measuring what you're trying to measure. And so randomized phase 2 studies can be significantly larger, although they're usually under 100 patients. A phase 2 study can go to more than 100 patients in certain circumstances, but they usually tend to be right around 100 patients if they're randomized. Now, a phase 3 study is the gold standard, and these are the kinds of studies that are used for FDA registration of drugs. Generally, they're designed to compare the new treatment against a standard treatment. Because there's a comparison involved, they tend to be larger because you're really looking to see, trying to get to the precision of that measurement of the difference between the responses in the 2 arms. They can be hundreds to thousands of patients. And the smaller the difference that you're trying to examine between the 2 arms, the more patients you need to be able to establish that. So, if you're looking for a home run, you can do a phase 3 study with maybe 100 patients, a couple of hundred patients. But if you are looking for a very precise measurement of something like a survival extension of 6 weeks, you probably would need thousands of patients for that. So it's kind of an inverse relationship. The smaller the difference you're trying to see, the more patients you need. So now I'll go into a little bit about how we develop protocols within SWOG. This is really specific to the NCTN INCORP SWOG setting, but I think that to some degree, the cooperative groups really established team science before team science was a thing. And we do definitely incorporate a lot of input from a lot of people during the protocol development process. The steps in protocol development, they look pretty easy, but they're not. It is a very involved and complicated process. Everything within SWOG, when it comes to protocol development, we work through our research committees. And so the responsibility of the committee chair is to really be the gatekeeper and to decide what kinds of things move forward and what kinds of things don't move forward. Anyone who is a member of SWOG can propose a study to a committee. They can come up with an idea and present it to the committee chair, and it may or may not move forward. Each committee has its own personality. So, there are some committees where the working group members vote on everything. There are some committees that have a request for application process. There are other committees where the committee chair makes all the decisions. So each committee works in its own way, but primarily the committee chair is the person who decides the go, no go for new ideas. Once the committee chair has approved a new proposal to move forward, then there's a next step where those proposals go to SWOG executive review. And the executive review committee members are comprised of our group chair, vice chairs, and executive officers, as well as some key administrative staff who kind of look at new proposals to really look closely at the feasibility of the study, the appropriateness for doing it within SWOG. The assumption is that the committee chair and the committee members who have already examined it have determined the scientific merit of the proposal. So really the SWOG executive review is looking at practical aspects of whether this study is appropriate for SWOG, whether we can do it successfully. And then the next step is that the proposal goes to the NCI. And for most disease sites, the NCI has an established external steering committee that takes a look at all of those concepts when they come in. And they make a decision about whether the research that's being proposed is of scientific merit, whether it's duplicative. We don't want to be doing research that someone's doing and we're just not aware of it. So the steering committee has a responsibility of kind of knowing the landscape of what kind of research is being done. If the steering committee approves a new concept for a study, then we can move forward with protocol development. That step 4 is looks like just another step in the process, but this is a really long and involved process. And it involves not only the scientific development and writing of the protocol document, but also contracting for drug supplies, making sure that we have things covered related to specimen submission. If we need Streck tubes, if we need specimen submission kits, all of those things have to be determined during the protocol development process. All of our protocols then go to a second step of NCI review where they look at the whole document, including the consent form. And we go through a process of NCI review. They send us a consensus review. We respond to the consensus review. And that may be an iterative process where we go back and forth with the NCI 2 to 3 times to make sure that the protocol document is as concise and complete as it can be. We make those revisions to the document, and then hopefully at the end, we get an approval. one of the challenges with doing this is that there are timelines associated with protocol development, and the NCI is pretty strict about this. The Operational Efficiencies Working Group several years ago, probably about 15 years ago, met and determined for the NCI that we really needed to make more of an effort to ensure that the science got to patients as quickly as possible. And even recognizing that there's a lot involved in developing a protocol document, the NCI still wanted to establish some definitive timeframes for when and how studies should move forward. And these are timeframes that are, there's a recognition that phase 3 studies are definitely more complicated than phase 2 studies, and that there should be a longer target timeline for phase 3 studies. So for phase 2s, we generally try to target opening a study from concept to activation within 210 days. And then there's also an absolute deadline of 450 days. I actually think that's shorter now, but where if we don't activate by the time that the NCI has given us for an absolute deadline, they can refuse to support the study. So even if they have approved the concept and approved the protocol document, they could withdraw funding support if we can't get it activated within that time frame. So there's a lot of pressure associated with that. In general, the concept approval timeline from submission to approval by the steering committee is about 2 months. The timeframe for protocol development, if everything goes as smoothly as possible, is about 2 months. And the timeframe between protocol approval and activation, which includes CIRB review, is about 90 days. So approximately 3 months. So a lot of times I think about a phase 2 study as being kind of a somewhat premature baby. And then a phase 3 study is a full 9 months. Phase 3 studies, our target is 300 days and our absolute is 540 days. And again, it's kind of the same thing, although the deadlines are a little bit extended because of the complexity of phase 3 studies. So in general, once a document is developed, you can understand that there's a lot of information included in the protocol documents. And I know you guys work with these all the time, but a lot of pages, a lot of information, a lot of things that seem to be boilerplate, but they're really not. And so we want to include as much useful information as possible. The key protocol sections we would want you to be familiar with when it comes to protocols you're working on. If a protocol is complex, so if there's a randomization involved, or if there's different steps, or patients enter into the protocol at different stages, then we will always include a protocol schema. If it's a very straightforward phase 2 single arm with no randomization and just basic registration, treatment, off treatment, then we wouldn't put a schema into the protocol. But anytime there's a divergence in the pathway for the patient, we'll include a schema. Section 5 for SWOG protocols is always going to be the eligibility criteria. So this is the details of what you would need to understand in order to be able to register a patient to that study. Section 7 is the treatment plan. And so for SWOG protocols, any information that you would need to be able to treat a patient on that study would be included in Section 7. And then Section 8 are the toxicities that you need to look for and what to do if there's a problem. So if there's dose modifications necessary, or if the patient needs to be removed from treatment for adverse events, or if a pause in treatment is allowed, how long can a patient pause treatment before they would need to be removed from protocol treatment? So a lot of those details are separated into those different sections. So if it's routine treatment and things like supportive care, that would be in Section 7. But if it's treatment that might be given only if a patient experiences adverse events, then that would be in Section 8. Section 9 is the study calendar. So we try to include as much detail as we can about the events that need to occur. When do laboratories need to be repeated? When do scans need to be repeated? How often do you submit specimens? Those kinds of details would be in the study calendar. Section 12 is a section of the protocol that we don't often use, but when we do use it, it's very important. So this is the discipline review section. So if you need to submit data that are not routine data that you would submit on in RAVE on the data collection forms, but if you need to submit something for a real-time review, such as a real-time pathology review or real-time radiation planning review or real-time surgical review, that's where that information would be included. And a lot of times those data are submitted to a different place and in a different way than your routine data submission in RAVE. So when you have that information in section 12, it's really critical to pay attention to that because it's not the run of the mill. Section 14 is the data submission section. So these, of course, reminders pop up in RAVE for the data that need to be submitted, but section 14 lays it all out for you. So you know these forms are due on these dates, or these forms are due when these events occur. More likely, you know, what forms need to be submitted at the end of every cycle, what forms need to be submitted when prompted by an adverse event, what kinds of forms need to be submitted at a patient's death. Those details are included in section 14. And then separate from the protocol document, we also do include a PDF of the master forms set. So you can actually take a look at all of those data points that you need before you're in the RAVE system, having to pull those out of the medical record. Protocol actions. So once a protocol is activated, there's a lot of things we send you. You guys get a lot of stuff distributed from SWOG, and those generally will fall into the following categories. So an activation will be the first time that you have access to a protocol. When we activate a protocol, it has already been through CIRB review, and all of the institutions that are part of the NCTN and INCORP are required to use the CIRB as they're reviewing IRB. But I know that a lot of your sites are also still very interested in reviewing the protocol documents. So even though the CIRB review is the official IRB review, there are a lot of individual steps at each institution, and it varies widely among institutions. We have some institutions that accept the CIRB review. They do a local administrative review and they activate the study. So some of our institutions can get a study activated within a day. Other institutions, it takes a lot longer. And it really depends on your local structures and who's involved in that process. An amendment to a protocol is relatively rare. They are very big changes to the protocol document that could impact on the risk-benefit ratio for patients. So it's the kind of thing that would potentially make a patient think twice about continuing on the study, or make future patients think twice about signing the consent form. So they are big changes to a protocol document. A revision is the more routine changes that you see on a regular basis, where we're updating editorial information, or we're trying to clarify something, or we get some new information that needs to be included in the document. So while they are still important and you need to know about them because they can definitely change the use locally, they're not the kind of change that might really prompt a reconsent discussion with a patient. A memorandum is new information on the protocol that doesn't actually go into the protocol document, doesn't affect the information that's already in the document, but provides some supplemental information. And this is the way we distribute external safety reports. So, you know, if we get these one-off safety reports from other studies that are being done that actually don't impact on the information in the protocol, but it's good information for your investigators to know, we would send it out with a memorandum. And then you'll also see temporary and permanent closures. A temporary closure can be built into the design of a study. And so it could be something like on a phase 2 study where we have 2 stages. And if we see a response in the first 15 patients, then we'll reactivate it for the next 15 patients. It can also be built into a study to look at safety signals. Or it can take place because of things we didn't foresee. If we have issues with drug supply, if we see safety signals that we weren't anticipating, then we would do a temporary closure so that we have time to evaluate what's happening with the study. And then a permanent closure, normally we always hope will occur only when we've completed registrations to the study and we've actually filled the accrual goal that the study called for. But we also will, on occasion, permanently close a study when we reach the point where we realize we're never going to finish this study, or if there's new information that comes out that lets us know that there's not a point in finishing the study. Someone's already answered the question, or other drugs are now being approved in the same setting. When we've reached that point of futility, then we would permanently close the study. This is the kind of information you can see on the SWOG website about a protocol. So when we activate a protocol, we create this protocol abstract that gives a lot of information about a study in a pretty concise snapshot. It's similar, but not exactly the same as the information that's included on the CTSU website. And we try to give enough information so that you can use the protocol abstract information as kind of a short guide to the protocol. So it gives you information about the title, how long has the study been open? What was the activation date? Who are the study chairs? There's a lot of items on this abstract page that are clickable. So if you clicked on the study chair's name, it brings up their contact information. And so you have a lot of ways of getting to detailed information that you might need to have. We also abstract out the basic eligibility criteria for the trial. And we include some publication information if there's been any articles published on that are specific to the trial. I do want to note for all of you that information that's contained in protocols is considered confidential. And the intent really is to make sure that you have enough information to be able to treat the patient on the protocol. So we want to get as much information out to you as we can, which often means we're sharing information that pharmaceutical companies see as confidential. And as part of your institution's grant with the NCI or your service agreement with SWOG, there is confidentiality clauses in that, but when it comes right down to it, we want to make sure that we are being as protective of patients as we can, and as protective of the scientific integrity of the study as we can be. So once a study has been published and it's completed, then there's not likely to be any impact in sharing that information. We've reached the point that it's not going to impact on accrual. It's not going to impact on data submission. It's not going to impact on data review. But until it reaches the point of publication, there is the possibility that any public disclosure of information could impact on all of those things: patient willingness to go on the study, the kinds of data that are submitted, the way that the investigators review the data. So we want to make sure that we're being as protective as we can be. I want to talk just a little bit about the National Clinical Trials Network, although that's not really the focus of SWOG's presentation, but we are a part of a network. The NCTN network is made up of several groups. And then, of course, we're also part of the NCORP network. The NCTN includes multiple other cooperative groups, including a a portion of the Canadian Clinical Trials Group, a portion of their support is provided through the NCTN. And of course, the Children's Oncology Group, we don't have that many opportunities to work with them, but we do occasionally, primarily because our studies, we are funded to focus on adult cancer research studies. But when it is convenient and possible, we collaborate with the Children's Oncology Group for adolescent and young adult studies, and then also for children's studies where the treatment is the same between children and adults. The network structure, I won't go into a whole lot of detail, but I think it's important to recognize that the NCI has provided a lot of services that are centralized, but each of the groups has their own support as well. So what you can kind of see, even though this diagram is a little busy, is that there are a lot of centralized functions, but then each group has their own operations, their own statistics and data management center, their own tissue banks, and their own participating member sites. And those don't always overlap. And particularly with the staff support with operations and statistics, those are entirely separate for each group. You should be familiar with the CTSU website, but if you're not, it really represents a lot of the centralization of the functions that the NCI supplies. Centralization of provision of protocol documents, of information about accrual, of safety information, posting of patient-facing materials and other documents. And then I would like to give you a little bit of an opportunity to ask some questions. If you have any questions, feel free to come to the microphones in the aisle. But if you don't, I'm happy to answer them at any time. My email is on the website, and I'm always happy to address any questions that have been emailed to me. Well, then, just to wrap up, I would like to introduce our protocol department group. I am the director of operations, so not only the operations office is responsible not only for protocol development, which is the piece that I'm presenting now, but we're also responsible for your quality assurance audits, tracking of your membership, meetings management, and several other operational aspects, including collection of conflict of interest information for your investigators. Crystal Miwa is our department head for protocol development, so our department manager. Laura Gildner and Mariah Norman are program managers for protocol development, so they supervise the other project managers in the protocol department. Patricia O'Kane is a senior protocol coordinator. She works with prevention studies and with cancer care and delivery. Katrina Morellis works with our melanoma studies, excuse me, as well as early therapeutics and rare tumors. Christy Klepetko is our GI protocol manager, and she also works with palliative and end-of-life care. Chrissy Laubach is working with our combo match studies as well as our industry-funded studies, SWOG CTP. Jennifer Beeler is our main project manager for LungMap. Sarah Cantu is our project manager for MyeloMatch, and some of you may be becoming very familiar with the new NCI platform studies. MyeloMatch is probably the next one to be activated. We expect it to activate this summer. Alicia Aranda works with our breast committee and also with some of our industry-funded studies. Sharon Palmer works with leukemia and myeloma. Chris Kipla is a jack of all trades. He is the project manager for our ImmunoMatch platform, and he's also been working very heavily on some of our LungMap studies. Kat Gassick, I don't have a picture of her, unfortunately, but she's our lymphoma project manager and also works with our industry studies. Megan Keim is our protocol manager for GU studies and for survivorship studies. Justine Trevino is now working with our lung studies and symptom management studies. And that's it for me. Again, welcome to SWOG, welcome to the group meeting. I hope you enjoy yourself and learn a lot during this time. And I'd like to introduce Michelle Counts, who's coming up next, and we'll be talking to you about data submission.
Good morning, everyone. Can you hear me okay in the back? Perfect. Thank you. I hope you're doing well. My name is Michelle Counts, and I am one of the DCs from SWOG Statistics and Data Management Center. And today I'll be talking about, I probably should click on my slide first. Good, data submission. So let's get started. Perfect. So as you may know, there are two data submission systems we'll be talking about today, the pre-rave on the CRA workbench and rave. The pre-rave is used for studies activated before April 2012, so it's been a little while. And right now it's only used for 4 SWOG studies. On the other hand, rave is used for all studies activated after April 2012, and there are 83 and counting SWOG studies. So let's talk about pre-rave first. So in the interest of time, we're only going to cover how to access a CRA workbench, how to look up patient charts, and we'll take a look at how the forms are organized. So if you have any questions about the pre-rave or CRA workbench, please reach out to your data coordinator. So first, I think I forgot a slide, sorry. All right, we are at the right place now. So to access the CRA workbench, please go to swog.org, log on using your credentials, and after clicking the member resources link on the top right-hand corner of the page, click on the CRA workbench link. And here we have the updated, beautiful CRA workbench homepage. So first, click on the patient management non-rave studies that you'll find to your left, and choose the first one from the list, which is data submission for non-rave studies, to search for your patients at your site. And from here, you can look up patients one at a time by their PID, which is your first option highlighted at the top right-hand corner. Or you can also generate a list of patients by searching the 3 categories highlighted above in the middle, which are study number, CTEP investigator ID, and also CTEP institution ID. And you can also apply filters as needed to narrow down your research. And as an example, let's type in study number S1007. And in this case, this is going to generate a list of patients on that study. And from here, you can click on the patient ID hyperlink to select the specific patient. And here's the page. The default tab that appears is the patient information tab, which displays the patient's registration information, vital status, and last contact date. And you'll also notice that this page will show notifications. For example, the red one in the middle, if you have any outstanding queries that you need to address. And at the top, in addition to the patient info tab, you'll see forms, expectations, and queries. They're very self-explanatory. The forms tab will take you to the forms used for data entry. The expectations tab will show any outstanding or upcoming expectations for this patient. And finally, the queries tab will show you a list of open queries for this patient. And that was actually it for pre-rave, very short and sweet. So let's spend the rest of the time talking about rave. So here is a brief overview of the topics we'll be covering today for rave data submission, which includes how to access rave, how rave is organized, data submission and amending data, and resolving queries, and finally, any resources that may be helpful for you guys. So first of all, when a site is approved for registration to a study in the CTSU regulatory system, people on the SWOG roster with the role of rave CRA will be sent an invitation to join that study. And I wanted to add that unfortunately, DCs like myself are unable to grant access to rave. So if you are not getting invitations, please contact your head CRA to make sure that you are set up as a rave CRA. And some studies require a delegation of tasks log, also known as CTL, which is completed in CTSU, and it identifies the CRAs and investigators that will be working on that study. And only individuals that are on the DTL can enter and edit data on the CRFs and rave. So please be aware that the DTL needs to be updated and sign up annually. So if you are unable to access or edit forms in rave, please check in with your regulatory team at your site. Now, this is the iMetadata portal to all rave studies you have access to, and this includes both SWOG and non-SWOG. On the right-hand side, you'll see the tasks section highlighted, which is where you find any invitations to join the study. And since this is my page, I'm not going to have any invitations, but if you do, it's going to look something like that green box floating to your right that says your study number, accept or decline. And underneath is the e-learning section, which lists any outstanding e-learning requirements. And finally, highlighted in the middle is your list of studies to choose from. And I really hope you don't have 521 studies on your list. I don't have 500, just make sure. And RAVE is organized by study, which means information can only be viewed for one study at a time. And this applies even if a patient is on multiple studies. So once a study is selected from the homepage, the tabs across the top of the page increase in specificity from left to right. So you'll see your selected study, your institution name, the patient ID, folders in that patient's chart, and finally the forms within that folder. So this is what it looks like once you've selected a study, and we are at the site level. And for this example, we've selected S1826 and a test site. And highlighted to your upper left-hand corner is the list of patients registered to study at the site. And to your right is the task summary, which displays outstanding tasks that can be viewed by clicking the arrow or the triangle next to each category. And since RAVE is very icon heavy, as you may know, there is an icon key available on every page highlighted underneath the subject list. And for all other forms, they're going to be towards the bottom of the forms. And next, we are at the patient page, where we call this the subject level. Folders containing eCRFs are highlighted in the navigation panel on the left-hand side, and highlighted in the middle are folders and their approximate due dates. And I wanted to highlight this, even though due dates are shown in RAVEs, as you can see, your expectation report and the protocol are always the best resources for determining when forms are actually due, especially as RAVE due dates may not always correspond to the expectation report due dates. And we would like you to follow the due dates on your expectation report. And task summary is once again highlighted on the right-hand side, but it's now specific to that particular patient. And finally, the add event drop-down box highlighted in the bottom of the page is a tool to certain forms that will not generate otherwise. For example, the lost to follow-up form. And the forms you see in RAVE are going to look a bit different from the paper CR versions. And also keep in mind that we can create conditional settings within RAVE that only show specific folders, forms, or fields, depending on how certain questions are answered. And the baseline tumor assessment form and the follow-up tumor assessment forms are examples of conditional forms. Highlighted at the top of the form, we have selected yes, does the patient have measurable lesions? And also yes, does the patient have non-measurable disease? And this is going to generate a target lesions and non-target disease forms in the same folder for baseline. As you can see here, since we have answered yes to both questions, on the left are the new baseline forms that have been added to capture that information that patient has both target and non-target lesions. And we also have a new follow-up folder that also contains target and non-target disease forms as well, highlighted to your right. So if you have only selected yes in the previous page for target lesions, then you will only get the baseline tumor target lesions form and the follow-up target lesions form. And in case you have questions about when or where to find specific document forms or forms in RAVE, we have created a RAVE form display document for every study, which is available on the master's form set found on CTSU. So if you are having trouble finding the master's form set, please reach out to your DCs. And I wanted to draw a special attention to the off-treatment notice, which is highlighted at the bottom of the slide, as we do get questions on how to find them in RAVE. So when the patient comes off treatment, you will need to trigger the off-treatment notice to populate. And this usually means amending the treatment form in the final cycle to indicate that the patient will no longer be continuing on protocol treatment. In this example, you would need to answer the question, will the patient continue to receive protocol therapy, with a no to generate the off-treatment notice. And this does need to be answered no, even in cases where the patient could be receiving another form of treatment, as here we are asking whether the patient is still receiving that specific protocol treatment that they have been receiving, which is usually no if they are generating or having generated the off-treatment notice. So let's take a look at a typical RAVE form. Field labels are to your left and field entries are to your right. And notice that some fields have the required units specified, for example, the height and weight in here. And next to the field queries or field entries are the status icons and edit icons, or we sometimes like to call them pencil icons. And you can open individual fields for editing by clicking the pencil icon next to the field itself, or open the entire form for editing by clicking the pencil icon at the very top of the form. And a question mark box next to a field label indicates help text that will generate data entry guidance and clarification. And most forms will have comments field at the bottom of the form where you can explain any unusual or unknown circumstances. And finally, at the bottom is the save and cancel buttons. So every time a form is saved, you will see the same static message instructing you to scroll down the form to make sure all your field icons are green check marks before assuming the form is completed. And once again, as a reminder, some forms will have fields that will not become visible until certain information has been entered and the form is saved. So let's discuss log lines, specifically, once again, our favorite, the baseline tumor assessment form in RAVE. And you can see right now that there's only one log line to start out with, and you can add more as needed by clicking the Add New Log Line hyperlink. And make sure you do save the form after you complete each log line so you won't be losing any of your work. And if you want to get rid of a log line, it can be inactivated by clicking Inactivate, selecting which log line to inactivate from the drop-down box, and then clicking the Inactivate button once again. And this does not actually delete the line from the table, but it strikes out the data on the log line. So you can also reactivate a log line in a similar manner. And it's important to notice that all active log lines that have been correctly completed have a green check mark at the very end of the log line. And if the inactivated log lines are, and the inactivated log lines will have gray inactive icon instead. And now let's take a look at the source documentation form. RAVE has a special field for the submission of electronic documents, and this form allows you to upload documents by searching for the file on your local computer by selecting the Choose File button to upload the document directly into RAVE. And also be aware that when uploading forms, the type of procedure selected might determine whether an expectation should be resolved or not. So please be sure to choose the most specific type and pay attention to the form instructions because they may instruct you on how to label certain forms depending on the study. And you'll notice that similarly to the form in the last slide, you can add in and activate log lines as needed. And before source documents are uploaded, you'll need to make sure that they are fully redacted and have the SWOG patient ID number added to at least one page of the document, and ideally the first page. And I personally suggest adding the SWOG study number and also patient initials on the form just to triple check that you are submitting the data for the right patient. And you'll not be able to edit documents once they are uploaded in RAVE. We do prefer PDF files, and there should be no special characters in the file name. And multiple documents shouldn't be combined in a single file, and each document should be uploaded to its own log line. So for example, if your study requires you to submit a CT scan and a bone scan, if you do combine those two files into a single folder and upload it, for example, under a type of procedure as CT, it's only going to resolve the CT scan expectation. So please make sure that you do keep them separate. And once the upload is complete, a link to the document appears along with the green confirmation check marks. And if you need to inactivate a log line, please be sure to remove the document from the log line before it's inactivated. And also be sure to read the instructions at the top of the form because they may vary across different studies. And a small FYI, when entering data onto a log line, it's important to use your mouse to select an option from the drop-down box to avoid non-conformant data error from populating. So please don't use your keyboard to complete the field like you would with the free text field if you are given the option to choose from the drop-down box menu. And as mentioned earlier, RAVE can be programmed to only show certain fields and forms depending on what data is entered. So let's take a look at an example. So this is one of the baseline forms asking for the patient's smoking status. And in this example, depending on the answer selected from the drop-down box, additional follow-up questions will generate below the question after the form is saved. So for question number 2, it asks, how long has it been since the patient has been smoking or last smoked a cigarette? In this case, we have selected less than 1 year to question number 2. So once the form is saved, a follow-up question generates underneath question number 2 with the new field to complete. And you can see here that the status icon is not green for the new field, which means it's not yet completed. So please be sure to save the form once the new field is completed and verify that all fields have green check marks. And here is an example of a sticky note. Sticky notes are used for reminders, and you just need to check the acknowledge box and save the form, or it will show up on your task summary until they are acknowledged. And as you can see here on the form, The presence of the sticky note does not affect the green check marks, and sticky note does not actually appear until the form is saved. So please be sure to do a once over after submitting the form and double check that the information is correct as entered and whether any additional documentation is needed to be included on the form. So let's switch gears to queries. System queries are automatically generated by Rave for edit check violations and non-conformant data, which is data that does not fit the field type, such as letters in a number field. On the left-hand side of the screen is an example of a system query that fired because the height field was left blank. And you can tell this is a system query because under the query, it says to open to cite from system. And another example of a system query is the non-conformant error that fired at the bottom of the form because the date entered, November 33rd, is invalid. And you can also see the red triangle icon for non-conformance next to the date itself. So to resolve this, we have entered a value for the height, and please make sure to check your units. This one's in centimeter, which grayed out the response box, meaning we no longer have the option to respond to the system query. And for any data change, you will be prompted for the reason you're changing the data. The options are entry error, new information, per query, and just choose whichever seems the most appropriate. It's not critical. There's no wrong answers. And now the bottom of every Rave form has a save without submitting checkbox. And checking this box will trigger a system query preventing data from being transferred to SWOG. And this will allow you to save the form but not submit any information. And however, unlike when you choose to save without submitting on the pre-Rave on the CRA workbench, edit checks still do fire in Rave. So we, the DCs, are trained not to look at forms and pretend they're not there until they are saved. They're saved by submitting. And as shown here is the query you can get when this form is saved with the box checked. And whenever you are ready to submit the data, simply uncheck the box and resave the form. And you can see here that all queries have been resolved and each line has a green check mark. And you can also see a tiny yellow triangle, which indicates that the data has been amended for that item. And you can click on the status icon of the field or that green check mark to see the audit trail if you wish to see who amended the data, when it was amended, and from what to what the amendment was made. And please note that all system queries must be resolved before the data can be imported to SWOG. So if you think you have submitted a form, but it's still showing up on your expectation report, please check that your form doesn't have any system queries that's pending your answer. And once it is imported, expectations tied to forms are resolved by the next business day. And other type of system queries or queries that you may know is your manual queries from yours truly, DCs. The main difference between system and manual queries is that even if you change the data, you also need to leave a response if a response box is present. And you can tell this is a system query or manual query, because it says to cite from DM data managers instead of from system. So let's address this manual query. So there is a response box, so you do have to enter response and if appropriate, fix the data. And here we have updated the date, and you'll notice that even though we did change the data, the response box still remains editable. And once again, this is a reminder that you have to type in a response. And we could have also typed in a response without changing the data in this case. And be aware that any data entered in the query response box is not saved to the database. Any information related to the query response and also to the patient chart should be included in the comments section or in the appropriate field on the form. And if you do find valuable information in the query response, your DC will send a manual query requesting you to type it in the comments section. And queries should not be closed until they have been addressed. And if you need clarification about what is being asked in the query, please reach out to your data coordinator. And once the query is resolved, all lines will have green check marks. The resolved manual queries will still show up on the screen, unlike system queries, which will disappear. And you can find open queries under the task summary. At the site level to the left of this screen, the task summary displays all patients with open queries. And on the right side of the screen, the task summary at the subject level displays forms that have open queries for that specific subject. And the second place that you can find queries is the query report from the CRA workbench. And now you should be able to see both manual and system queries. I think it used to be just manual before. And highlighted above is an example of what manual queries look like on the query report. And a very helpful feature of this report is that you can actually see which Rave field and form the query is on, so you'll be able to find it in Rave. And even though you can't see the queries here, they must be addressed in RAVE. And finally, here's a list of RAVE resources available for you from the CRA workbench. And you can still continue to use the CRA workbench as a reporter for all things related to patient management for SWOG studies. You just can't submit or amend data here. And your front line for technical support with RAVE is the CTSU help desk, and their contact information is here. And it's also available through a very, very teeny tiny customer support link on the bottom of every RAVE forms. And that concludes the presentation. So if you have any questions with the particular study, please reach out to your data coordinators. And thank you for your time and attention. Can I take any questions at the moment? All right, if not, you have our distribution list to contact us and or call us. And I would like to introduce Monica Yee, who will be presenting on patient-reported outcomes next. Thank you.
Good morning. As Michelle mentioned, my name is Monica Yee, and I am a Program Director for Cancer Control and Prevention Studies in Seattle at the Statistics and Data Management Center in Michelle's office. And I'm going to talk to you today a little bit about patient-reported outcomes. Today is just going to be a very brief overview of the what, the why, your role, and resources and training available to you for patient-reported outcome reporting. Patient-reported outcomes are instruments that evaluate the functioning and health outcomes from a patient's perspective. They're incorporated and included in some treatment studies and most cancer control and prevention studies evaluating new treatments for interventions and treatments. I already mentioned that. They're reported by the patient and they evaluate the health and overall status without interpretation by a clinician. You might hear different terms such as quality of life, health-related quality of life, patient-reported outcomes. This diagram is by Dr. Lynn Henry, who's our SWOG Symptom Control and Quality of Life Committee co-chair, and tries to demystify a little bit about what all those terms mean. So in general, quality of life is a very general term that can mean different things to different people. It's sort of like the word beauty. It's way too general and amorphous to be measured. Health-related quality of life is a multi-domain concept that represents the patient's general perception of the effect of illness and treatment on physical, psychological, and social aspects of life. Patient-reported outcomes can be defined as any report of the status of a patient's health condition or health behavior that comes directly from the patient, without interpretation of the patient's response by clinician or anyone else. Self-reported patient data provide a rich data source for outcomes, and this definition reflects health-related quality of life, including functional status, symptoms and symptom burden, pain and fatigue, for example, and health behaviors such as smoking, diet, or exercise. So patient-reported outcomes captures critical information for SWOG trials, such as outcomes reported by patients, which can be different than those reported by clinicians or researchers, as I mentioned, especially when there is evidence that side effects are underestimated by clinicians, as done in research in 2006 by Dr. Ethan Bosch. Benefits of including PROs may lead to improved communication, satisfaction, and symptom management. Your role in PRO research is very critical. You are the primary for quality PRO data collection and submission. So please be prepared for this important role. Complete training on the online SWOG Pro training module before administering questionnaires for any trial. Be familiar with the pros and process for each study. Perform quality control of data and patient completion prior to submission to avoid inconsistent or incorrect data, and submit data in a very timely fashion. Consider how you would navigate any potential patient issues or barriers before they're encountered by referring to the protocol for guidance. Study-specific details about PRO administration Data collection forms, you'll locate those on the CTSU via CRB approved documents. CTSU website sometimes is a little daunting. So definitely contact us if you have any questions about where to find any of these forms. The time points and windows for completion and study-specific instructions are also included in the protocols as well as on data collection forms. The way the questionnaires are administered for the protocol may be various ways, or you might see combinations. Sometimes the forms are collected via hard copy on paper. Most typically, we are using that mode of collection right now. There's also electronic data collection, and we're doing that in one of our trials right now, a cancer control, symptom control trial, S2013, that many of you are probably participating on. And then also on some of the cancer control studies, the data is collected by a third-party collaborator by phone or other method. Here's some of the sections of the SWOG protocol. You'll find pro information. There are many of them, and the information is embedded in many of those sections for eligibility, treatment. Section 9, the study calendar, as you've heard today, is a very important resource. And the data submission information, the data submission sections 14 tells you when to submit the data. And then section 15 includes many of the details that are for the instructions of administering the instruments and how to deal with different situations that they come up when you're working with your participants. And then section 18 describes what the objectives, the background, and what the instruments are to give you a little more information about the reasons we're collecting the data on this particular trial. There was a SWOG Pro training module, as I mentioned, and it describes the what, why, and details of how the Pro administration is to be administered to the patients. It's about 20 minutes. It's narrated by Dr. Jamie Myers, and it's located on SWOG.org under clinical trials, protocol workbench training, patient-reported outcome section. And here's how you would navigate to that area. There's so many parts of the SWOG workbench that even I'm still discovering. So there's an overabundance of information on there, and one of them is including the training for this really important role. And here's just a screenshot of the training. We're not going to go over that today. This is just a very brief overview of what's included for you. And if you have any questions about any of the administration of the questionnaires, where to find forms, please contact Cancer Control Question at crab.org. That is also the email address for any questions for cancer control questions in prevention or cancer control. And now I would like to introduce Phyllis Goodman of the Statistical Center, who is going to be describing reports. You'll learn all about the reports.
Good morning. I have the pleasure of being the last speaker before break, which means I know you're anxious to be done, and we're running a little late, so I will try to get through this without too much ado. So I'm Phyllis Goodman. I am a statistician at the SDMC. I'm the person that works with all of the performance measures and the reports that go out to your head CRA and PI about quality. So to talk a little bit about what we mean when we talk about quality, when we support our, when we submit, our grant, the NCTN grant to the National Cancer Institute, which is going to be in the later part of this year, what they ask us about is timeliness. Were the required data forms submitted, and were they submitted on time? For timeliness, like I said, for accuracy, other than chart reviews that are done with auditing or monitoring, we don't compare the data on the submitted forms to what's in your source documents. But what we do provide is accuracy on whether or not the submitted forms are free of queries. So back in the 1980s, so pre-rave, we developed something called the expectation system. And I heard Michelle talk a bit about expectations, and maybe you're not aware of what those are. But expectations identify the submission requirements of forms, reports, specimens, patient completed forms, and anything else that the protocol requires. Originally, when this was developed back in the 1980s, there were 3 expectations, and they were for the on-study forms and follow-up. But it's expanded over the years so that right now we have over 700 different kinds of unique expectations. So an expectation is anything that's expected to be submitted to the Stat Center, and it reflects the protocol-specific data and submission requirements. And that's my old dog waiting expectantly outside our coffee shop. But expectations can also be posted conditionally based on a treatment arm, a stratification factor, a disease status, or an answer to a consent question. So when are expectations posted? They can be posted at the time the patient's registered or during protocol treatment and follow-up. So at patient registration, the kinds of things that we're looking for are the baseline requirements. The forms, the on-study form, the baseline abnormality form, source documents, the path report, specimens that are required at baseline, and other study-specific requirements such as the quality of life questionnaires or imaging or other kinds of source documents. It can also, at the time of registration, you can post expectations for things that when you know they're due at a defined point in the future. So a sample due at week 4 or a quality of life form due at week 8 sort of thing. So it's the defined point that's known. And those kinds of expectations that are generated at patient registration appear on the confirmation of registration. But they can also appear during the course of the patient's treatment and during follow-up. These kinds of expectations are what we call dynamic expectations. And for treatment trials, typically the forms that are included that we call dynamic are like an adverse event form, a treatment form, or follow-up tumor assessment form. But there are other study-specific kinds of forms as well, such as a concomitant med form, some kinds of lab values, some kinds of, I think on the lung map studies, there are other kinds of study-specific forms. These expectations are dynamic in that they're time period specific, and the posting of them depends on the patient's actual visits, when they actually came in. The due date's determined based on the data submitted on the prior forms regarding when the patient was last seen, the time period that's being covered, and when the next visit is due. The submission of the data is based on data we have stored in the SWOG database regarding the expected visit and assessment times as defined by the protocol. Other expectations that can be conditional are things that we call conditional because an event has happened. So the patient's progressed, so we expect new kinds of things, or they've relapsed, or they've gone off treatment, so new things are posted. Another kind of expectation, the current guidelines for expectations are we follow the CTSU guidelines for when expectations are due. And currently from the CTSU, it's the 15-15-30 rule, which means baseline data is typically due within 15 days of registration. On-treatment data is due within 15 days of the visit or the event, and off-treatment data is due within, I think I didn't update this slide. Did I not update this slide, Sam? I didn't update this slide. It's 15-30-60. So I realized I have to update this. So it really is on-treatment data is due within 30 days and off-treatment data is due within 60 days. But because you can't trust me, always be sure to check section 14. Because data submission timelines also for older studies, these were the older guidelines, and even older studies might have things slightly different. Specimens, both baseline and post-baseline specimens, you need to check section 15 for due dates, but typically blood products, urine are due within 15 days, except for those kinds of tubes such as Streck tubes that are due the day after the specimen was collected. And tissue, we typically give 30 days, recognizing that getting to your path lab or sometimes where the tissue is collected isn't at your institution, but somewhere else. So we give a little bit longer for that as well. Another kind of expectation we have are what we call vital status updates. So here we're really looking to see whether or not the patient is at least being contacted. And this is continued follow-up is expected while the patient's receiving treatment and after they're off treatment as well. And this is also dynamically posted based on that last time that the patient was seen. And is protocol specific. This is just an example of a typical treatment schedule where, for the first 2 years, patients treated every 3 months, we would expect forms regularly. Then once they go off treatment, the follow-up is a little less frequently, maybe every 6 months. And then by year 4, maybe only annually. So when we're calculating our vital status update expectations, we're looking at what we call time since patient's last contact. And we'll calculate that as the days between the day the report was run and the day the patient was last known to be alive, their last contact date. So on our older studies, the last contact date, and older is really only pre-2019. Last contact date was updated from a number of forms, including the treatment form, the adverse event form, the follow-up tumor assessment form, the SWOG follow-up form. And then we went through and gleaned the database and looked for what is the most recent date. And that became harder and more confusing. It was more work for sites that were putting that same date on multiple forms. So we changed it. And starting in 2019, we created what's called the Vital Status Update form. And it is now the wave of the future. We are in the future, 2023. So I guess I can get rid of that too. That we have only a vital status form that looks like this. And all it has is two questions. Was the patient last contacted? And what was the date of that last contact? But the most important thing about this form is it needs to be the very first form that you submit when you're entering data in RAVE. And that's because it sets a date within RAVE that says this is the patient's last contact date. And if you try to submit a form from a visit that has a date later than what the RAVE thinks is the last contact date, you'll get a system error. So by submitting this form first, you relieve yourself of future system errors. So what do we have in terms of reports for quality? We have an expectation report, which I'll talk about, the institution performance review report. I'll also touch on the query report and the ineligible patient report. Accessing the report, similar to other things, you navigate to the SWOG workbench via swog.org. And the link is down here, the patient reports and data quality. And that's the list of the kinds of reports that you'll see. So when you get to the expectation report, this is a snapshot of what it will look like. I have a section at the top that's news. And in the news section, it's, I put things there that are relevant to who's ever looking at the expectation report, things that might have changed about posting the expectations for something or specimens are changing. So that's in the news section. And then, you can navigate to the bottom of the page where you will get to the actual reports. So expectations reports, we have two different versions of them. There's a monthly report, which is static, generated at the beginning of the month, the second of every month. We also have, and this is the report that is sent to your head CRA and to your site PI and is used for calculating site metrics. So that's kind of the monthly snapshot. But we also have a current report, and this is dynamic and it reflects the data in our database up to the time that you run that report. So if you've been submitting a lot of data and you want to know, okay, is that expectation report reflecting that data that I submitted? You can navigate to this page the next day, because as Michelle said, it takes a day for the data to make it into our database and see whether or not those expectations have been resolved. Both of these are downloadable to Excel. So that you can manipulate them if you're happy. I like working in Excel using the filters, using the sorting, using highlighting to see what I need to do. And they also both identify expectations that will affect your compliance statistics. So the expectation report doesn't just show you what's overdue, it shows you what's coming due, but also for those overdue things, it says, okay, it's overdue, pay attention, but these are the things that really matter. And you can identify those. This is a snapshot of what the current report looks like. The filters at the top of it look like. I think you can filter on a patient ID. So if you're in charge of certain patients or if you're in charge of certain studies, you can filter on that. But you can also filter on this section here, which kind of tells you about the expectation categories, which I'll talk about in a little bit. And it helps you know what those things are that are really counting against you. So a current expectation report will look like this. All the necessary fields. I'm highlighting there what a dynamic expectation looks like, because that's the one that's time period specific. And you can tell because it has that, that time period beginning on that date. This column here shows you these things that are affecting your IPR metrics, and I'll explain what those codes mean in a little bit, but you can then see which are the ones that, I really better get on that because my head CRA is going to be bugging me about that form in particular. Some of the other fields there's the due date and also the days overdue. So you can see here that if something doesn't have any days overdue, that means it's due in the future. Expectations looking for updated follow-up have kind of different flavors depending on when those were created in our timeline. So just a follow-up expectation looks like that, and it might just be an older study looking for a follow-up form. Whereas this follow-up, the one below it, is looking for a very specific off-treatment vital status form for a very specific period. Other dynamic expectations, as I showed you before, an adverse event period beginning or a treatment form period beginning. and then a specimen submission, what it will look like. And it will tell you the kind of specimen we're expecting and then more details about it. You know, what time point it was expected or the kinds of tubes that it was expected to be pulled in. And a specimen expectations can get very sticky and they're hard because the specimens have, it used to be, I always talk about the old days and people are tired of me talking about the old days, but the old days, there used to be one specimen that was ever submitted. And it was a specimen for confirming that the patient was eligible for the study, that it was the right path. And that was it. There were no blood specimens. There were no urine specimens. There are no subsequent blood and tissues, but it's gotten a lot more complicated as our science has gotten more complicated. And as the kinds of questions we can ask of the kinds of tissue we collect or specimens we collect. So they're complicated. And specimens expectations, first of all, patients can opt out of expectations. There are some that are required for the protocol, and you'll see that in the eligibility criteria, that they're absolutely required. Patients can't opt out of those. But for those future TM kinds of studies, patients can opt out of specimens. And if they've opted out, those expectations won't appear. And if you see them and you think your patient's opted out, you need to contact us to figure out what's going on. The other thing is that baseline specimens are used both for assessing eligibility or screening or stratification or future endpoint determination. And they're also used for banking or future translational medicine studies. Post-baseline specimens can be posted at a fixed point, as we talked about before, posted at the time of registration, or can be posted based on events such that the patient progressed, so it's posted conditionally. When you're going to log a specimen in, and I believe that this will be talked about more in later, but how I want to refer to it in terms of expectations is we've modified this page very recently. You have to choose a filter at the top because that list got so long and so unwieldy. We thought we need to force people to use it because they weren't using those. I mean, if you just choose the lab, you're going to still have a pretty long list of specimens. But if you go back and choose not just the lab, but a time point, then you'll have a much more focused list of specimens that you're choosing from, because a lot of them sound the same. What tube is a little bit different, or maybe the time point's a little bit different. So by using those filters, you can really focus in on what you need to be submitting. And then the last thing is if you have a patient who was supposed to come in at week 3 for a specimen and they never came in, and you didn't collect, or the patient said, no, I'm willing to do specimens in the future, but this one, I don't feel good today. I don't want to do it. So you can't collect it. How do you resolve that expectation? You go to the specimen tracking, which will be talked about later this afternoon. And there's a link on the left-hand side that says notify that specimen cannot be submitted. And when you use that, it will resolve the expectation. So now I've talked a little bit about institution performance review, or IPR. And what IPR is, it's our statistics that we use to monitor and measure how well an institution is doing in terms of compliance to the data submission requirements of a protocol. Are those forms being submitted? The compliance guidelines are outlined in policy number 33. I got something here. It's run monthly and contains the compliance rates or percentages for each category. And it's posted on the SWOG CRA workbench, but also mailed to the head CRA and PI monthly. But you can always get to that IPR report. So when we talk about IPR, there are 4 different categories that we'd look at. There's initial forms. So those are expectations associated with the patient registration, the data. It includes specimens that are needed to evaluate eligibility, treatment or stratification assignment, or a future, and specimens that are used to those categories. We also have a category called post-baseline form submission. So everything that isn't included in that initial form set in terms of forms. We have that vital status updates. Are you keeping in touch with your patients? And the specimens that are not included in those initial form sets. So specimens that are kind of for those future TM studies. The overdue items contributing to those different categories are identified on your expectation report, as I showed before with the IPR column. And the categories and definitions are also on the expectation and IPR report. So when you're looking at a current expectation report, if you look at that IPR column, you can see these are the things that are counting. So that follow-up form isn't just overdue, it's 355 days overdue. It's going to count against your site. If it was 3 days overdue, it wouldn't count. There are guidelines about how overdue it needs to be. In terms of what the report looks like, here's a report. It shows you the 4 different categories with 2 subcategories, meaning initial form set. We look at registrations within the last 13 months, but we also say if anything's more than 13 months overdue, that's really bad. So it has its own category. Same thing with specimens, more than 13 months overdue, its own category. And then an asterisk indicates which metric is out of compliance. So talking about compliance levels, what we've done is each of the 4 different compliance pieces have a different denominator because they mean different things. So when we're looking at initial form set, we're looking at registrations. So one patient can be registered on a study, might have 3 registrations. There's the potential for that one patient to be overdue 3 times for each of those registrations. So registrations is the denominator. And a site is out of compliance if more than 10% of the registrations are more than 30 days overdue. And we mean that all of the items that are associated with that initial form set must be received. So a single overdue item will result in that whole registration being called overdue. When we're looking at post-baseline forms, we're looking at forms as the denominator. And if more than 5% of the forms are overdue, or more than 60 days overdue, then you're out of compliance in that category. And vital status of more than 15% of patients, because here one patient has multiple registrations, maybe on multiple studies, but we're looking at patients. If more than 15% of patients are overdue, more than 60 days overdue, out of compliance. And for post-baseline specimens, it's more than 10% of specimens are more than 30 days overdue, out of compliance. And that as opposed to a report card, rather than hoping for 100%, here you're aiming for zeros. Okay, so not a report card, you're looking for 0. In terms of consequences of poor performance, if it's 2 months in a row, 1 month, we kind of let, everyone has a bad month. If it's 2 months in a row, then the institution, an email comes from me to the PI and head CRA, kind of as a warning saying, we've noticed this is going on. You need to pay attention to what's happening here. If it's 3 months in a row, then it gets a little more stern, like you are actually at risk of suspension if your site is out of compliance for any one of the metrics for more than 3 months in a row. And it means that you might not be able to register patients to future studies until that IPR issues are resolved. I'm also just going to touch briefly on query reports. This isn't really my area as much, but I do think it's important. Michelle talked about queries, and those are the kinds that we're talking about. The queries posted by the stat center, the data coordinators, the central monitors, the QA team, the biorepository based on their review of the samples that they get. Rave queries do appear on our reports. They're in a separate section. They're separated out from the kinds that are posted by the stat center. And all queries are currently not tied to institution performance review or expectations. So query report is different than an expectation report, but we are looking at creating it a separate new compliance measure as a component of future performance. Query report is located on the CRA workbench, and it's as showed here. Another report is an ineligible patient report. Now, eligibility determines whether the patient will be included in our primary analysis or maybe our secondary analysis, or when we're analyzing the stats or analyzing the data, kind of knowing why someone's ineligible is important. So we really want sites to check their ineligible patient reports. It isn't part of a performance measure because it's kind of a hard thing to get a handle on. Differing studies are really challenging in terms of their eligibility criteria. Sometimes they're written better than other ones. But we really want you to review why patients have been coded as ineligible, and this is why. So we have 3 different categories of ineligibility. So we have something called N, meaning patients clinically ineligible. But check, is there a data entry error? Maybe the data coordinator is looking at something and the value is supposed to be less than 20, and you've entered 71 rather than 17. So check your data, make sure that, oh, I messed that up. I can fix that, and that patient will become eligible. Another kind of ineligibility is what we call insufficient information. Here, eligibility can never be confirmed because the material wasn't available. The scan wasn't done, or a specific lab wasn't done. So from our information that we have, there's nothing we can say that they're ineligible, but we also can't say they're eligible because we're missing that piece of information. But then we have a third category, which is reverse NR, or reversible. Here, there's missing information. It's reversible. They might be ineligible, but if you submit that lab because it was done and you just didn't write it on the form, then that patient can be made eligible. So the ineligible report looks something like this, accessible once again on the CRA workbench, has the filters at the top as all of our studies do, and it gives you the patient ID and the code that was used and the reason. And so you need to look at that because maybe there are things that you can actually fix. And then the last thing I'm going to just talk about briefly is the CTSU reports. So these are reports that are generated for RAVE studies. And the data quality, it's called the data quality portal, accessible on the CTSU website. It has a link for delinquent forms and queries from that are posted by the data coordinator, as well as the system and non-conformant queries. It includes all our SWOG RAVE studies, but not our pre-RAVE studies. And it also includes other network group studies. Michelle mentioned, they aren't exactly the same. The systems were developed differently under different kinds of guidelines, different kinds of rules. And our metrics right now are really based on what's in the expectation reports. So if you're ever wondering how come something's not showing up on the CTSU report, but it's showing up on your report, you need to work towards our report because that's really what's used. Couple of just quick facts. The auditors do use the reports that we generate, but they use them on a case-by-case basis. If they know they're going out to a site, they just might ask us, are there any issues going on? It isn't part of their formal audit report, but they do have access to it. The NCI and other network groups, do they have access to our reports? Currently, these reports are only used by us and the information is not routinely given to them, but if they asked us, we probably couldn't say no. Once again, if the DLs in terms of who to contact, if you have any questions about this, the expectation report, this is the DL to use. And just, it's hard getting all this data in. Things are complicated, old studies, new studies, what kind of queries, what kind of expectations, but all of that is for creating the most high quality studies we can. And that all requires your effort. And we really appreciate everything you do and a really hard job. So thank you very much. And only 1 minute over. So if there are questions, but my guess is you want to head out. Great. So I think it's a 15-minute break. Is that right? Yep. I'm getting the thumbs up. 15 minutes.
Okay, we're going to go ahead and pick back up with our next set of presentations. I'd like to go ahead and introduce Connie Susponic here to talk to you about long-term follow-up and the importance of that for you as a CRA.
Well, hello, everyone. It's an honor to be here. It's a little scary seeing all these lovely faces. It's so good to be back together again, not remote. So welcome to San Francisco. My name is Connie, and I've done this work for a bit, since the 90s, really. And I'm the chair of the ORP committee. So we are your committee. If you need anything, we're the Oncology Research Professionals Committee. Let us know what you're needing, what you're not finding, et cetera. But our goal is to help you get what you need to do your job every day. This topic is near and dear to my heart. I think sometimes our focus is so much on accrual because we have to accrue to meet our goals as a program. And many of you are new. You might be coming into a caseload of patients that you didn't enroll yourselves. They've been on study a long time. And sometimes I feel like long-term follow-up takes second fiddle to the core work of what we do. And yet it's so, so, so important. So we're going to talk about what it is, why it is, how it is, some of the rules of the road, and go from there. Long-term follow-up is really answering questions. And it's our patients' stories. They've entrusted us to tell. And always think of it as that. You're telling the story. So if it doesn't make sense to you, it's not going to make sense to SWOG, to NCI, or to the sponsors. Our goal is to collect the information and send it in so that the whole thing makes sense and is complete. And so answering questions about, did their protocol treatment get completed, discontinued? Do they have any toxicities? Have they had them? Are they now resolved? Are they responding to therapy? Sometimes these things play out over years and decades. Some of our studies are until the end of life. So telling the story completely and fully is really important. And as you'll hear from Mike LeBlanc later on, every patient matters, every time point matters, every milestone matters, because our studies and the integrity with which they're developed depend on our ability to tell the story. We're going to come back to this, but I want to point you to SWOG policy number 30. If you haven't really looked at the SWOG policy manual, and I confess I'm one of those people that hates to read policies, but if you don't read any other policy, read this one. This has so much detail and it's only 3 pages long, but it's kind of like the truth about what you have to do when. And it defines the responsibility for patient follow-up, unique scenarios where patients move from one place to another, and criteria that gets used for all the above. So why do long-term follow-up? It's good for our patients, it's good for the studies, it's good for us. It assures continued medical surveillance and makes sure that our meaningful end results are reported appropriately. It helps us capture accurate data about what happened with this patient, their disease, their survival. Also, sometimes there are late effects or other things that could happen, and this helps us pick those up as well. For SWOG, the follow-up intervals are pretty set. It's generally every 6 months for at least the first 2 years and then annually after that, once they move into long-term follow-up. Section 14 of the protocol is really important. There's a lot of sections in the protocol. There's lots of bits and pieces and appendix and this and that and the next thing. But when you're trying to figure out how to map out the course of your patient's journey, be sure to look at protocol section 14.0 for when your data is due. Always read the protocol carefully to find out how long you need to follow up people. Don't make assumptions. Just go by the protocol. It's your source of truth always. Patients on some older studies may be followed until death. And if you're not sure, you're in doubt, go with the most conservative interpretation and you can't go wrong. It's always easier to not have to have something later than to have missed something that you really needed. The important thing as you manage all of this, and it's one thing when you start out and you have 1 or 2 patients and you meet them and you know them and you remember them vividly. But as time goes on, you might have 200 or 300 patients that you're following. And pretty soon it all starts to blur together. So it's really important that you have a how for tracking. And there's no one right answer for how you do this. It's just you have to do it. So figure out something that works for you. Generally, you want to track by the date of the last contact. Use the expectation report. We have a lovely tool that we get from CTSU called DQP that we love and we hate, but generally it has some dates due on there. So keep an eye on any reports that you get. And whatever you use, I know there are people that still use index cards, sticky notes, clinical trials management systems, deluxe computer programs, paper in a folder, sticky notes. It really doesn't matter what you use as long as it's efficient, it works for you, and you remember your patients and when they're due for follow-up. Make sure if you do it, you write it down. That becomes critically important. The date of the last contact is important for vital status. The date of their last clinical assessment or disease assessment, if there's any progression, recurrence, subsequent treatment, and especially watch for any reports of new cancers. If there's anything that could be related as an adverse event, those are important things to listen for, watch for, and make sure they get documented. Places to look for data, the hospital record, treating physician record. It could be their family practice office. It could be that there are letters or notes that you have from other family members or in their chart. It's really important to understand that every patient counts, but they all have the potential to be lost. Life happens, and the patients themselves aren't lost. I kind of hate this word. We say lost. Patients are lost to follow-up. We're the ones that are lost, losing them. They're not lost. They generally know where they are and what they're doing, but it might not be connecting with you when they need to. So be proactive. Generally, it starts at the beginning. And ideally, as you grow in this role, you'll have a chance to build a relationship with patients and clinics. But assume things are going to happen. I mean, we had COVID, we didn't plan on that. How many patients have we had a hard time keeping track of during COVID? Getting to know them and their journey will help you keep track of what's happening for them, sharing with them what comes next. But confirm and update contact info at every visit. Verify the plan and the timeline for their next follow-up and build in handoffs. So preventing gaps, preventing that distance that can happen. Collecting demographic information is one way to keep track of things. And this can be found in the patient chart or their EHR. You want to know their patients, sometimes the patient's demographics. Sometimes referring physicians can be a good source of truth. Relatives, numbers, cell phone numbers. One tool that I've seen sites use, and we used to use this in our prevention trials, but it was a participant information sheet. Sometimes going over this with a patient just helps kind of visually remind them, we're trying to keep track of you. We're here for you. We hope to keep you in our thoughts and prayers and in the loop of communication. It's two-sided, it's two-way, because our pledge to them is that we're going to keep them apprised of how their study is unfolding, not just because we want something from them. So as things are going forward, it's really important that we know how to reach them in case something changes, something happens, there's an update we need to get to them. And generally, patients are very willing to share this with you as their research coordinator. We often talk about this on our team that, things happen, things change. Clinic staff have turned over heavily, as have our research teams. But at the end of the day, it's often the research coordinator who's the glue in their journey, sometimes almost in a navigation-like role. You may be the one who's there with them when they're diagnosed, when they see the surgeon, when they see med-onc, when they see rad-onc, when they go to primary care, when they go into follow-up. So keeping in touch with one another is important. This will be in the slides, and so you can steal this liberally if it's helpful to you. These are just ideas for data to capture so you can reach them later. But communicating regularly is pretty important, and it's important to have that relationship and be part of their journey. If you have a chance to be part of their informed consent, treatment planning, be at those visits with end of treatment, follow-up planning, key time points, and foster good relationships. Sometimes it's as important to keep track of the patient, but also the office team. Make sure they know that you're there and what your role is. So fostering good relationships with the physician's office personnel, the front desk, the receptionist who call and say, hey, your patient's here. What do we need today? Health information personnel, the cancer registrar, navigators, genetic counselors. There's lots of people that interface with our patients. So making sure they're part of our journey and part of the team, and understand the patient's commitment to the clinical trial, and engage them in helping to support and celebrate the patient's journey. Building a bond with the patient, stopping by when they have appointments, birthday cards, notes, appointment reminders, giving them postage paid envelopes. If there's something they have to share back with you, it's easy. Make it as simple as it can be for the patient to keep in touch with you, as well as you keeping in touch with them. There's some other tips we're going to share. We won't go into great detail, but they're going to be in the notes. But if somebody's lost, in other words, you can't find them, sometimes looking in the EMR, the electronic medical record, social media is another thing. If it's public domain, we can find lots of information sometimes. And something as basic as they're still alive is pretty important in the survival data calculations that are part of the trial. So these are some ideas for where you can get public domain information. There's a lot of internet resources listed here that you can Google. And I'll just leave these and you can look at them in the slides. Some other lists of things where you can look. There's lots of places to look. Internet resources for Social Security Death Index even. So I want to come back to this policy number 30 and talk a little bit about the responsibility for patient follow-up. This can be found on the SWOG member site under policies and manuals, policy 30. You can also get to this same place with the policy manuals from the CRA workbench, which I think you're finding and hearing about today. But I want to highlight some of the words in this policy. All institutional and individual participants in SWOG are responsible for the follow-up of all patients registered by the institution and or the individual at the institution for as long as the patient remains alive or for a protocol specified length of time. The commitment to patient follow-up remains regardless of the funding status or membership status within the group. It's really important. So even if you're coming into this new and it isn't your responsibility, or it wasn't originally, you didn't enroll these patients, your site wasn't involved in A, B, C, X, Y, Z. But if they're on your watch now, they are your responsibility. And it's really important that there be an all-in commitment to make sure that we take good care of them. It's a good time to look back at this policy when there's been a change. Maybe your site's really struggling with funding and they're closing down. Or maybe another site closed down and you're absorbing their patients to help support them. Maybe the docs have changed. Maybe the patient's moving from one SWOG institution to another. Maybe they tell you, I don't want to do this anymore, and they withdraw consent, or you can't find them. Those are all good times to look at the policy because definitions matter. And if you put in the wrong definition or you count them in the wrong pew, so to speak, lots of other things start happening. Like, for example, metadata rave. What a nightmare. You accidentally turn on the wrong button. You're suddenly going to get hit with queries up the wazoo that you can't ever escape from. So it's important that you use the right termination, the right definitions, and make sure you put things in correctly. Let's start with a simpler one, which isn't that simple at all. Patient transfer. So the patient goes from one institution to another. It's important to know that you as a CRA or oncology coordinator can often and rave click some buttons and things can happen. But this shouldn't happen without lots of people being aware of it. And make sure you work with your program leadership. There are things that have to be in place throughout your system. You need to contact the new site for the transfer, make sure they even have the study open and that they're active and in good standing with their IRB. You do initiate some patient transfer forms online. It's important that you resolve all your expectations and queries before the transfer takes place, and you provide the accepting institution with a copy of their research record and case report forms. The accepting institution, so if you're the one receiving a patient from another site, you complete the form indicating acceptance and make sure you have IRB approval before conducting any study activities. And then you need to work with your regulatory team and compliance folks on your team to make sure that you're in good stead with the consent form that's signed for your institution and your HIPAA authorization. So some things have to be in place before we can just say, great, you can go see that doctor over there at that other clinic. There's some really important things. Maybe the patient says, I just need to stop. I'm too tired. I'm worn out. I don't want to do this anymore. It's a really significant time to have a critical conversation. You want to try to always leave the door open for patients, honor their wishes. We don't need to coerce them into staying on because it's better for us or better for the study. We need to do what's best for them, but you want to leave the door open as much as possible for them to return to the trial if their life changes or things start going better. So you verify with the patient, is your wish that you no longer get treatment? Or would you rather just say, I don't want to be followed by the protocol, or both? Sometimes they just need a break. Withdrawing consent to participate in a study does not necessarily mean the patient also withdraws consent to be followed. So make sure that they understand that they have that option to stay connected with their research team and their research site, even if they do take a break from their treatment for a bit. Before you finalize the status for consent withdrawal, again, check the policy, look at the definitions, find out what documentation has to be in place for this to happen, and make sure your program leadership is aware that you have a patient withdrawing consent. There are folks that are tracking things like regulatory and IRB linkages and how many patients are in study, because if an amendment comes out or some safety notice, we have to be sure we know who to notify. Know and understand the implications. For example, what about their specimens that are stored? Future research permissions on their consent. There are things to go through. And I would encourage you to talk to your study coordinator at SWOG to verify how to document this and be sure they're in the loop with you, especially if it's the first time you've ever done this or had to document this so you don't miss anything. Sometimes too, there's the situation that is considered lost to follow up, which is a little tricky too. There have been times where there's a lot of pressure to make sure your IPR, your institutional performance report, or your query report gets cleaned up. And it has been a temptation at sites to say, well, I can't find them. I'm just going to mark them lost to follow up and get my query reports cleaned up. Please don't ever do that. It's a terrible idea. Doesn't help the study, doesn't help your patient, doesn't help you in the long run. There are some really strict criteria to meet before this can be the word that we use and apply. If it's been more than 2 years since you were last able to reach them and you have not been able to contact them and find them in any way, ideally, you want to try to reach them at least 3 times in 3 different ways to reach them, like a certified letter, a phone call, multiple attempts. Document everything you try to do before finalizing the status. Again, review the policy and make sure you're using the right definition at the right time, and that you've documented what needs to be documented. Again, inform and discuss with your program leadership, connect with your SWOG coordinator, make sure you're doing it right. Know what your local site requires you to do as well. And then update your local files with the status change. And if possible, anything you can do to leave the door open for the future is always better. There is a specific form. This is what it looks like. It's on the CRA workbench under patient management. And at the top of it, you'll see it says, has it been at least 2 years since the last patient contact? And what was that date? And then what have you done? How many attempts have you tried, et cetera? A couple other things for long-term follow-up. SWOG has uniquely created what's called a long-term follow-up protocol, where studies that are really a long ways out can be moved underneath the umbrella of this protocol. It relieves the burden for local IRBs doing continuing review for lots and lots of studies. That way they can do continuing review for one study umbrella that includes a lot of older studies that are underneath it. So if you're in that situation and your site has a lot of those older studies, take a look at this. Again, it's posted on the CRA workbench, and there's a list of those studies under reports and study management. There's a list of no follow-up required studies as well. At some point, we're still maybe potentially analyzing, but there's no further follow-up required for those patients that are a long ways out, and they're called no follow-up required studies. Check out the CRA workbench under reports, and it will tell you follow-up is no longer required on which studies, and it includes a date about keeping the records. And you want to keep things until the SWOG date or institution required date, whichever is longer. These are going to be older studies, and it's hard when you come in and if you're new to the program and these things happen 10 years before you ever began, tracking them down can get a little tricky. But use the SWOG website and the definitions to help you with that. This is what it looks like on the CRA workbench. Study reports. And there they both are. Those are the two bullets, the two buttons that you want to look for those two topics. It's really nicely done. And SWOG is one of the research bases that has really put a lot of work into helping consolidate those kinds of activities. So it's not so much long-term work that doesn't help you achieve very much other than busy work and regulatory. So in closing, just realize that our patients have entrusted us with being part of their journey. You have a really, really important role. Telling the story really does matter, because if we leave out the pieces, we don't capture the whole story, it impacts ultimately the journey that they've been on. They've contributed to this trial, which is really an important treasure that they've given to the whole world, actually, because we're changing standard of care forever. And you are in a very unique position to make sure that what they do gets captured accurately, timely, and that their data matters. So that concludes my presentation, but I'm happy to take questions if there's a few minutes for that. Well, then I just wish you all the best and welcome to the family. We need all of you. There's been such a shortage of research coordinators. It's delightful to see you all here, whether you're new to this role or new to SWOG. Thanks for being part of this journey.
Great. Hi, everybody. My name is Gabby Herbert. I'm a data coordinator at the SWOG Statistics and Data Management Center in Seattle, and I'm going to be going over adverse event reporting for SWOG clinical trials. So in this presentation about adverse events, or AEs as I'll refer to them, I will be going over some definitions and background information about adverse events, pointing out where you can find relevant information about them in SWOG protocols, important components of reporting adverse events, and how to report adverse events using electronic case report forms. We'll start with some definitions and background information on AEs. First, what is an adverse event? The SWOG CRA manual defines an adverse event as any unfavorable and unintended change in a patient's condition from the day that protocol treatment began, regardless of cause. An AE might be new, meaning it was not pre-existing before starting study treatment. It might be a pre-existing event that recurs with increased severity or frequency, or it might be something that was present at the time of study drug initiation, which was exacerbated following study drug administration. Another key term to define is toxicity. A toxicity is an adverse event caused or possibly caused by the drugs or treatment used in the study rather than a reaction to cancer or some other underlying disease. You might see both terms used in SWOG protocols depending on the context. However, for patient assessments and reporting, we collect all adverse events, not only toxicities. Here are some examples of adverse events that might occur when a patient is on study. Note that some of these would be considered toxicities. That is, some of them are potentially related to study treatment and some are not. However, all of them must be reported. Excuse me. All of them must be reported given that they occurred while on study. Yes, even including abnormal labs and a wrist fracture. Because adverse events are temporally associated with the subject's participation in research, they must be thoroughly documented and reported regardless of clinical significance or attribution to study treatment. Another important term to know is serious adverse event. Sometimes a patient will experience an unexpected or severe reaction to protocol treatment. These adverse drug reactions are called serious adverse events or SAEs. Maggie from the operations office in San Antonio will go more into detail about SAEs and how to report them after this talk. So as background information for adverse events, it's important to note that there are two types of adverse event reporting for SWOG trials. There's expedited reporting, which is done through CTEP errors and will be covered in the next presentation. Then there's routine reporting, which we'll focus on in this presentation. Routine AE reporting is the reporting of all AEs, which are captured on case report forms in metadata rave. So why is it important to collect routine AEs? We always want to consider the safety of our experimental regimens as well as effectiveness. This is why evaluating frequency and severity of AEs is almost always a clinical trial objective. Accurate collection of adverse events is also statistically important, as inaccurate AE reporting may bias trial results. And on this slide, you can see some examples of trial objectives that are related to adverse events. So now that we've covered some definitions and background information, let's discuss a key resource for AE information, the SWOG protocol. Here's a summary of the sections of the protocol that provide useful information regarding adverse events, and I'm going to go over most of these in a little more detail. And although not technically part of the protocol document itself, the master form set is really useful for finding trial-specific information about adverse events. Protocol Section 3 includes information regarding study agents, for example, storage requirements, stability, administration, and supply information. Section 3 also provides a list of toxicities that are known to be caused by the agents used on the study, including a list of toxicities that do not require expedited reporting. Those toxicities are presented in the specific protocol exceptions to expedited reporting, or SPEAR, shown on the right-hand side of this slide. Section 8 is the toxicities to be monitored and dose modification section. As you can see, it contains a lot of important information. It lists certain toxicities that may be observed on the trial, as well as symptom management medications and adverse event reporting requirements. Section 8 will always tell you which version of the AE criteria is being used for that study and where to access it online. All newer SWOG protocols use CTCAE version 5, but older protocols might use CTCAE version 4 or even version 3. So it's very important to check Section 8 of the protocol to see which version is being used for that protocol. Another item this section details, sorry, I got ahead of myself. There we go. Another item this section details are the dosage changes that might be required during treatment should a patient experience an adverse event. It's very important to review this information in Section 8. If Section 8 dose change instructions are not followed, the patient might receive too much or too little protocol treatment. Not only would this be bad for the patient, but it could also skew the toxicity profile on the study. So it's extremely important to follow the dose reduction and/or re-escalation instructions described in Section 8. If you ever have questions about dose modifications, please direct them to the study chairs. Their contact information can be found in Section 8, as well as the title page of the protocol and the protocol contact information page at the beginning of the protocol. So let's look at an example of dose modifications. Required dose changes are sometimes displayed in a dosage modification table within Section 8. In this example, the dose levels are listed in the top table, but you might also see dose modifications expressed as a percentage of a previous dose. The lower table shows dose modifications for the possible AE of renal impairment. On the left-hand side of the bottom table, adverse event grades or the severity of an AE are noted. We're going to cover grades in more detail a little later in this presentation. You should always use the highest grade that was experienced during a treatment cycle to determine the dose modification. In this example, for grades 1 and 2, there's no change in the dose level. For grades 3 or 4, however, the dose is decreased by 1 level. So for example, if the patient experienced a grade 4 renal impairment while receiving the full 1,000 microgram per day dose, after the patient's renal impairment resolves to grade 2 or lower, the drug would be resumed at the next lower dose, noted here as dose level -1 at 750 micrograms per day. The Section 9 study calendar outlines activities and timeframes and is a useful reference and scheduling guide. Please note that it is not a comprehensive resource for study adherence and does not replace the information in other protocol sections, so be sure to review all sections carefully. AE assessments are denoted in the study calendar in the toxicity assessment section, noted in the blue box here. Each X represents a required assessment time point. An AE assessment should be performed at the end of each cycle reporting period and prior to administration of the next planned dose. Do note that because some treatments can cause long-term or delayed effects, some protocols may require you to continue to assess toxicities even after the protocol treatment is finished. Most frequently, this is required for a minimum of 30 days post-treatment or until the patient starts treatment on a new drug. Generally, AEs occurring after that 30-day post-treatment window are only collected if they're severe and at least possibly related to protocol treatment. These are referred to as late effects or late adverse events and will be defined in the protocol and on the case report forms. Just very briefly, Section 16 addresses ethical and regulatory considerations, and in older SWOG protocols activated through 2019, AE reporting requirements were also presented in Section 16. Finally, the master form set, also known as the all forms packet, is a great tool for understanding the AEs on a trial. It is a PDF containing all case report forms or CRFs for a particular trial. The master form set is found under the protocol related documents tab in CTSU. Note that the adverse events CRF will be reported electronically via metadata rave, but the paper versions are available if you want to use them as a worksheet. Here's an example of the paper versions of an AE form. These forms are a good resource to see what data elements are required to be reported for each adverse event, as well as protocol-specific instructions and AEs of interest. So for example, you might learn on these forms if the study is collecting start and stop dates for each adverse event. So now that you know what toxicities and adverse events are and where to find that information in the protocol, I'm going to talk about how you should report these events to us at SWOG. First, I'll discuss the criteria that we use to grade adverse events, which is called the CTCAE. To accurately capture adverse events, all SWOG studies use the Common Terminology Criteria for Adverse Events, also known as the CTCAE, for all adverse event reporting. The CTCAE are a standardized criteria that define the severity or grade of a specific event. They provide a uniform method of grading AEs. There have been several versions used on SWOG trials. Version 5 of the CTCAE was published in November 2017 and is used for SWOG protocols activated after that date. And as I mentioned earlier, older protocols might still use version 4 or version 3, so be sure to check section 8 of the protocol for which version is being used. This is a page from the CTCAE version 5, where we can see how CTCAE terms are organized and how the grades are displayed. As we see from the bold title at the top, we're looking here at the immune system disorders section of the CTCAE. CTCAE terms are organized into system organ class sections like this, with the exception of the investigations section, which covers adverse events captured by lab tests. Within system organ class sections, the CTCAE terms are organized alphabetically by adverse event term on the far left column, with definitions provided below each term. Version 5 also includes navigational notes, which can guide you to other appropriate terms. For example, allergic reaction, noted at the top left of this slide, has a navigational note to use a different AE term if the allergic reaction was related to infusion. Additionally, each AE listed indicates possible associated grades, which range from 1 through 5, as well as a clinical description of that adverse event at each grade. I'll talk more about grades shortly. CTCAE terms can differ from typical terminology. I've listed some examples in blue. On the left, we have common clinical conditions, pneumonia, thrombocytopenia, and shortness of breath. And on the right are the correct CTCAE terms, which you would report on the case report forms: lung infection, platelet count decrease, and dyspnea. The document search feature, Control F, can be extremely useful to find the most accurate term if you're using an electronic copy of the CTCAE. If a suitable term cannot be found, you might need to use the term other within the appropriate CTCAE category. However, beware that other should only be used as a last resort. I know when I review AEs on charts, I check AEs reported using other against the appropriate CTCAE guidelines. And if I find a potentially more suitable term, I'll place a query with my suggestion for your consideration. Okay, so now that I've warned you to use other sparingly, I'd like to highlight a time when the other category should be used, which is when reporting a COVID-19 infection. For any COVID-19 infection, an adverse event must be reported on the adverse event or late effects form using the CTCAE term infections and infestations other, and then using the text COVID-19 in the specify field. I'll show you an example of this in a few slides. Any other AEs the patient experienced must also be reported, such as dyspnea or acute respiratory distress syndrome, and SAEs must be reported via CTEP errors as applicable. Some of you might recall at the height of the COVID-19 pandemic, all SWOG studies collected COVID-19 diagnosis data on the COVID-19 diagnosis and deviations form. As of June 2022, this requirement ended for most trials. However, the trials listed at the bottom of this slide continue to collect the first positive COVID-19 test on the COVID-19 diagnosis form, in addition to routine and expedited AE reporting. I briefly mentioned grades earlier. CTCAE grades provide clinical descriptions of severity for each AE. Here you can see the general CTCAE grading criteria, but as we'll see on the next slide, there are specific clinical descriptions of grades for each specific AE, and not all grades apply to all AEs. Here's another page from the CTCAE version 5, this time from the blood and lymphatic system disorders section. Note that each AE has grade descriptions specific to that AE. Specific descriptions of grades could be lab-based, but some are more clinical. If you look at the far right column, you can see grade 5, if applicable, is always death. You can also see here that not all AEs have all grades. Febrile neutropenia is a good example. If a patient is noted to have febrile neutropenia, they automatically have at least a grade 3. Eosinophilia, on the other hand, only has 2 possible grades, grade 1 and grade 3. When in doubt, always refer to your CTCAE guidelines for grading. In addition to grading adverse events, many studies require reporting of an attribution code. This is not part of the CTCAE, but is something that most trials request. An attribution code is a description of the relationship of the adverse event to the entire regimen of protocol treatment. Attribution codes range from 1 to 5. 1 and 2 are considered unrelated, and 3 through 5 are possibly to definitely related. Please note that the attribution codes must be assigned by the physician, not the patient or by the CRA. Some SWOG studies also collect status codes in addition to grade and attribution, which represent the state of the adverse event at various points throughout the study. The status can be new if it was the first occurrence of the event on the study, or for previously reported AEs, the status could be continuing at the same or lower grade, or increased in grade, or improved and worsened, and that is in comparison to the previous cycle, or for cycle 1 in comparison to baseline. Here are some of the other data items that might be collected for each AE. Recall that the adverse event form is available on the master form set and provides insight on which specific data fields are being collected for a particular study protocol. So just some general rules for AE reporting. First, to ensure accuracy, record events in real time as they occur. Trying to remember adverse event details later on might result in errors or underreporting. Please also be sure to report all adverse events, not just those considered related to study treatment. For most studies, report only the most severe grade since prior AE assessment. However, please refer to your AE form instructions for your specific study, as there are some studies with exceptions to that. Please also avoid using other if possible. If you can find a term in the criteria that more accurately describes the event, report using the exact term. And lastly, when in doubt, reach out. Questions about routine AE reporting can be directed to us at the SWOG Statistics and Data Management Center. All right, so now that you know about what's involved to collect data on adverse events, I will go over how to actually report adverse events in our SWOG electronic data capture systems. SWOG currently has two systems in which trial data is entered, the CRA Workbench and Metadata Rave. The CRA Workbench is only used for legacy studies, so I'm not going to cover it in detail in this presentation. If you want more information, the online version of this training course includes additional slides about reporting AEs in the legacy system, or you can contact the studies data coordinators. Now I will go over how to report AEs for studies that use Metadata Rave for electronic data capture, which includes all studies that opened after April of 2012. The first part of reporting adverse events in RAVE is to complete the adverse events assessment form. You want to indicate the adverse event reporting period start and end date, indicate whether the patient was assessed for AEs and whether the patient experienced any AEs. And lastly, indicate the date of the most recent AE assessment. The Cycle 1 AE form will capture all events occurring up until administration of Cycle 2 study treatment. In order to capture all AEs occurring during a treatment cycle, the reporting period must include the following cycles: day 1 pre-treatment lab assessments and physical exam findings. Thus, the date of most recent AE assessment should always be at the end of the reporting period, ideally on day 1 of the following cycle. When you answer yes to whether the patient experienced AEs, this will generate an adverse events report form. Once you hit save, the adverse events report form will generate in the list of forms in the cycle shown on the left-hand side of the RAVE screen. For newer studies which use CTEP errors integration, you will also see the expedited reporting evaluation form generate in the left-hand panel. RAVE CTEP errors integration will be discussed more in the SAE presentation after this presentation. I'll focus now on the adverse events report form. Here's what the adverse events report form looks like when you first open it. For studies that do not use CTEP errors integration, you can edit the log lines directly on this page. But for studies like this one that do use CTEP errors integration, you'll need to click the pencil icon to open the first line for editing. Here's what the log line looks like when you open it for editing. There's a banner near the top which tells you which log line you're working on, and the data fields for the log line are below. As I mentioned before, the individual fields on this form will vary from study to study, but will include the CTCAE term, grade, and attribution. The first thing to do is to select an appropriate CTCAE term. Always use the arrow to select the CTCAE term from the drop-down menu. You can use the text field to search for the term you're looking for, but be sure to still select the term from the drop-down. Even if you type the term exactly correctly, you'll get a non-conformant data entry error if you do not select the term from the drop-down menu. After selecting the CTCAE term, you'll select the grade from the drop-down menu. Here's another reason it's important to use the drop-downs. Only the applicable grades will display for each term selected. For example, here, only grade 1 through 3 displays for the AE term cough because those are the only applicable grades for cough in CTCAE version 5. After selecting the grade, you'll select the attribution, again using the drop-down options. You'll then complete the rest of the fields on the form, which again will vary by study. For studies with raised CTEP errors integration, you'll need to select the applicable seriousness criteria for the integration to work. After you save the form, you'll be taken back to the logline view where you can see the AE you just entered. You may enter, excuse me, you may, beg pardon. So you might enter further AEs one at a time by clicking add new logline. Conversely, you can use the inactivate link to inactivate any AEs that were entered in error. Sorry, I've got a frog in my throat. Okay, though I mentioned other terms should be used sparingly when they must be used, as with COVID-19 infection, you must use the provided text box to type in further details. This is a free text field, not a drop down, where you'd enter a very brief description of the AE. There's a character limit on the specify field, but if needed, you can elaborate further in the comment section at the bottom of the form. Here's an example of what happens when you type in an entry instead of using the dropdown. I typed in pneumonia instead of selecting lung infection from the dropdown. So I got a non-conformant data entry error indicated by the pink highlight and the red triangle icon on the log line. So how do you amend a log line when you've made a mistake? First, you click the pencil icon on the applicable log line, which will open that line for editing. In this example, since we've entered non-conformant data, Rave has already highlighted the fields that need to be amended and opened them for editing. If that were not the case, you could click the pencil icons on the fields that you need to amend. First, amend the AE term by deleting your entry and then selecting the correct term from the dropdown instead. After correcting the AE term, you must then select the correct grade from the dropdown. Remember that the grades are specific to each AE term. So if you correct the AE term, you must also reselect the grade. When you save the form, it'll take you back to the logline view where we can see that the corrections made have resolved the non-conformant data. For studies like this one, which use CTEP errors integration, the final step will be to run the expedited reporting evaluation using the form highlighted in yellow on the left-hand side of the screen. Doing so will fill the SAE report recommended column on the right-hand side of the screen. This step must be repeated anytime you add or amend a logline on the AE report form. You'll learn more about the expedited reporting evaluation form in the next presentation on SAEs. So that concludes my talk about AEs on SWOG trials. If you have any questions, please feel free to reach out to us at the SWOG Statistics and Data Management Center. And I have a minute here if anybody has any questions now. Hi.
I have a question about when you talked about people that were put in, like, you have no pistol, which you a question's come up. Well, they also had dyspnea, fever, chills, etc. And within the lung infection, that's all parts of it. Are we expected to also include all of those other symptoms of that emergency event?
Yeah, good question. Yes, all AEs should be reported. So anything that the patient experiences would need to be reported separately. Any other questions? Back for more.
So I'm thinking of another study where it's known that they would have a cytokine storm. And the definition of cytokine storm talks about the fact that they would have hypertension, right, chills, etc. And it wasn't a SWOG study, but in that particular study, they wanted to only report cytokine storm, not going to report all of those other adverse events or symptoms per se that are adverse events. Would SWOG still agree with, you know, we have a definition of what an adverse event is? They have that written at the bottom of our CTCAE. It'll give you a definition of what this adverse event is, and if it's contained within that definition, then we still have to report all of those other adverse events that
Yeah, good question. If it's excuse me, if it's included in the CTCAE definition, then I think you'd only need to report that one term. Okay. Anything else? 3, 2, 1. That's my time. If you have questions, feel free to reach out. Thank you.
Hi, everyone. I'm Maggie Spillers with the Operations Office in San Antonio, and we'll be discussing SAE reporting. Fair warning, this is pretty dry material, but you do get a break after this. When I first started working with SAEs, I thought finally something that's black and white in research. I was wrong. It's not black and white. There's so many nuances, so many gray areas. So hopefully we can clear up a little of your questions here, but if not, I'll be repeating our contact information a lot. So if you have any questions, please email us anytime. That's what we're here for. And here we go. So the first thing that you would want to know, and if you don't write anything else down from this presentation, write down our email address. It's at SWOG.org. We're here to help you answer any SAE questions. If you're not sure whether an AE needs SAE reporting or you're having technical issues, contact us first. Chances are it's something we've seen before and we can help you faster than any of the various help desks that you may reach out to. SAEs are a subset of all AEs collected. Just a reminder that SAE reporting is always going to be in addition to routine AE reporting, not instead of. And as Gabby mentioned, the CTCAE is the common terminology criteria for adverse events. There is a grading scale which refers to the severity of the AE. And the CTCAE has grades 1 through 5, but it is important to note that not all A's listed will have all grades. And this is a repeat of what Gabby just presented, but just a reminder, grade 1 is considered mild, grade 2 moderate, grade 3 severe, grade 4 is life-threatening, and grade 5 is always death. When you're talking about the attribution of the AE, you're talking about the relationship to the protocol treatment. There's two larger categories of attribution. The first one is unrelated and the second is related. And within each of those overall categories, there are subsets, subcategories within the attribution as well. And the overall category unrelated also contains unrelated and unlikely. And then the overall category of related could be possibly, probably, or definitely related. Where do you find the information about reporting SAEs for SWOG protocols? Either Protocol Section 8 or Protocol Section 16. Section 16 is being phased out, but you still will see it in some of our older protocols. But for the most part, you'll mostly find SAE reporting information in Protocol Section 8. This is an example of an SAE reporting table for investigational agents. It's probably hard to read from where you are, but the important thing to note is at the very top, you'll see a list of 6 criteria. This is referred to as the seriousness criteria. An AE has to meet one or more of these criteria to be considered an SAE. In the middle, you'll notice the timeframes for reporting. So if an AE met one of those 6 criteria, how quickly do you need to report it to us? And that's the section in the middle. The sections at the bottom refer to SAEs that may happen after the patient has finished protocol treatment, and there are some specific instructions there. If you have trouble interpreting this table or you're not sure where your patient falls, email us at adr@swog.org and we'll be happy to take a look. This is an example of an SAE reporting table for commercially available agents, drugs that are already approved by the FDA for this indication. We're not as concerned with SAEs for these agents, so the requirements are a little more loose. You'll notice that you only even have to think about reporting SAEs if they're grade 4, grade 5. And for grade 4s, they need to be unexpected, which means not listed in the list of known side effects, at least possibly, probably, or definitely related. And all grade 5 events would require SAE reporting. In some of our protocols, you'll also notice additional reporting requirements. So the first arrow points to an AE of special interest, something that the pharmaceutical company or SWOG wants to capture in addition to the typical AE reporting. So in this instance, any thromboembolic event, any grade, regardless of attribution, is always going to be an SAE for this trial. You may also notice some exceptions to SAE reporting listed, and the second arrow points to those exceptions. So for example, because up to and including grade 4 myelosuppression is considered an exception, if a patient has a grade 4 neutrophil count decreased, that's not ever going to be an SAE because it's specifically excluded here. However, any questions, email us anytime. That's what we're here for. We are happy to take a look and give you our interpretation. The SPEAR, Gabby did touch on this just briefly in the previous presentation. This is very confusing to many people, myself included. But the simplest way to think of the SPEAR, which the SPEAR stands for Specific Protocol Exceptions to Expedited Reporting, and it's that green column all the way on the right. The simplest way to think about it is it's just a list of AEs that will never require SAE reporting. So for instance, your patient has grade 2 abdominal pain with hospitalization over 24 hours. In most cases, that would be an SAE. However, since it's listed on the exceptions list up to and including grade 2, that's not going to be an SAE. Now, if they have a grade 3 abdominal pain with hospitalization, yes, that is going to be an SAE because it's not specifically excluded. Some protocols may have more than one investigational agent, and so there may be more than one SPEAR in the protocol. If that's the case and you find conflicting information, always use the lower of the grades to determine SAE reporting. And again, email us or call us if you're not sure. Something important to remember is that the SPEAR only applies to SAEs. It does not ever apply to routine AEs. So think of the S in SPEAR as serious. Don't worry about the SPEAR if you're just reporting your end of cycle AEs that you've collected. Reporting deaths, most SWOG protocols will require death to be reported while a patient is on protocol treatment or within 30 days of stopping. There are a few different CTCAE terms that you can use to report death. It's always best to try and attribute that death to a CTCAE term. For instance, grade 5 cardiac arrest or grade 5 multi-organ failure. If you cannot find the cause of the death, you can use death not otherwise specified. Or if the patient died very suddenly and you just don't have any idea what happened, you can use sudden death not otherwise specified. Or if the patient passed away from the natural course of their cancer diagnosis, you would use grade 5 disease progression. Pregnancy reporting, luckily this does not happen very often, but if you do have a patient that becomes pregnant while they're on a trial, you do need to always report that as an SAE. In addition to the SAE reporting, there's a specific form that NCI requires for all pregnancies, and that's linked here. It's just a simple form that you fill out by hand and fax to NCI. The terms that you would use to report a pregnancy is going to be pregnancy if the actual study participant is pregnant, pregnancy loss, or death neonatal. Something that is often confusing is secondary versus second malignancies. First, we'll talk about secondary malignancies because these are the ones that are required to be reported as SAEs. So secondary malignancy, you can also think of it as a treatment-related malignancy. So a patient with breast cancer got chemotherapy, and a few years down the road, they have leukemia caused by that chemotherapy. That's going to be considered a treatment-related malignancy or a secondary malignancy. And we do want to know about these. We do ask that you report all secondary malignancies as SAEs. These are not going to be metastasis of the initial cancer. It's going to be a new cancer that was directly caused or possibly caused, at least possibly caused by treatment for the existing cancer. In contrast, a second malignancy you can think of as a new primary. Patient has prostate cancer and they are a farmer and they've now have a basal cell carcinoma on their ear. It's pretty clear that's not going to be related to their prostate cancer treatment. It's a new primary, a second malignancy. These do just require routine AE reporting and rave. So this is a huge topic, how to report an SAE, and I cannot cover it all here. It's not an easy process, but I'll go through a few things and then just want to drive home the point that please call us. We will walk you through this over the phone. It usually takes an hour and a half to report your first SAE if you've never done it. It's complicated. We'll do a screen share or a Zoom call, whatever you need to help you get this submitted. For older protocols, you'll actually report directly in CTEP errors. But all of our newer protocols use something called the rave integration with CTEP errors, and it is very complicated. So please just let us know if you need any help. It's exactly what we're here for. There's additional information in these links for resources to help you navigate the RAVE CTAP Errors integration. One of the important things to know is that the integration uses really basic rules. To the best of my knowledge, it thinks that every grade 3 and above AE should be an SAE, but we know that's not the case in real life. So if you do get a recommendation in RAVE and it's not correct, follow the protocol. If you still have questions, call us, email us, we'll help you interpret those. And like I mentioned, the recommendations can often be incorrect, but SAEs should always be reported based on the protocol. The recommendation in RAVE that you'll see on a screenshot here in just a moment is just that, it's a recommendation. It's never going to be a mandate to report. Also, the opposite can be true. RAVE may not recommend an SAE report, but you know this is an AE of special interest and it always requires SAE reporting. So again, that's an instance where you would ignore the recommendation in RAVE and report based on the protocol. If RAVE recommendations do not match the protocol and you're not sure which is correct, email us, send us a screenshot, whatever information you need to provide, just let us know and we'll be happy to take a look and see how we can help either confirm that it does or does not require SAE reporting. And if you are in a report, an SAE report, and you find a field that's grayed out, you cannot change it in CTAP errors, that means it's being pulled directly from RAVE, so you would need to change that data in RAVE. This is an example of what the expedited reporting evaluation form looks like. This is the form that provides those recommendations in RAVE, yes or no, do I need to report this as an SAE? The first arrow is pointing to the little box that you'll check to send all AEs for evaluation. The second arrow is you can change the recommendation from create to none. If you've determined, no, this doesn't match the protocol, I don't need to report this, feel free to change that to none. You can override it that way. And then the third arrow is the link to actually go to CTAP errors and create your SAE report. This is what the recommendation looks like if you're in RAVE. So this patient had a grade 3 dyspnea, and it did require hospitalization. So RAVE says, yes, this does need to be reported. Most likely this is true. This does need to be reported as an SAE. However, always check the protocol or email us if there's any doubt. The expedited reporting evaluation form does always have to be manually run anytime you change the data on the AE report form. If you've determined, oh, it's actually a grade 4, not a grade 3, you're going to need to rerun that evaluation so that if you did need to report it as an SAE, it would be pulled over into the CTAP errors system. This form you can think of as the link between RAVE and CTAP errors. The recommendation is going to be shown in the middle of the page, but again, remember that they're not always correct. The 3 possible recommendations that you'll get in RAVE are going to be none, create, or amend. Amend, you'll see if you've already created an SAE report and you have changed the data and it thinks that that SAE report should be updated. It's going to tell you to amend the report, and you'll click that same link to go ahead and create an amendment to the original SAE report. Just a reminder, anytime you update that form, you've got to manually run the expedited reporting evaluation. If you do have pop-up blockers, most of the time we can help you get around those. But if it's something that's really locked down by your IT department, you may have to put in a help desk ticket internally to use the integration because it does require pop-up windows to work. Also noting that the recommendations are dynamic. So if you've already reported an SAE because you got a recommendation of create, it matched the protocol, then the recommendation will change to none once you've reported the SAE because there's no further action required. Nothing needs to be done right now. So don't be alarmed if it did say create and now it says none, or if it did say none and now it says create. These are dynamic fields based on what's going on with the AE report form. Another thing that we do, the SWOG SAE team does look at all SAEs reported by your site, your institution, prior to each SWOG audit. So we will send you a list of SAEs that we have noted as being reported late on our end. This does not mean they were truly late. A lot of times something happened on a Friday, it was a holiday weekend, you didn't find out till Tuesday. That's okay. We're not going to consider it late. We just need to know when you found out about it. So if you can, go ahead and put your date of discovery of the SAE in the SAE report itself. But if not, or if you forget or you're not sure, we do give you another opportunity before each audit to reconcile any SAEs that we've marked as late on our end, but may not be truly late. Do I need to report the SAE to my IRB? The answer is it depends. If you use a local IRB, which is generally going to be for older SWOG protocols, you need to consult your IRB to see what their reporting requirements are. If you use the central IRB through NCI, which is for all new SWOG protocols, almost no SAEs are going to require reporting to the central IRB once you follow the algorithm and realize, no, this didn't cause great harm. It was not unexpected. So follow the algorithm. But if you're not sure how to interpret the algorithm, give us a call or send us an email at adr@swog.org, and we'll be happy to walk you through that. The next few slides, we've got some frequently asked questions that we receive. And Gabby did go over this, but what does expedited reporting mean? All that means is SAE reporting. It's completely interchangeable with SAE reporting. You'll see both terms used in SWOG protocols. I'm not sure if this AE requires SAE reporting. Should I submit a report just in case? We see this a lot. We get a lot of reports that we end up withdrawing because they did not meet the requirements. And you're frustrated because you spent 2 hours on this and you've redacted documents and we tell you, oh, we're just going to delete it. So please call us, email us first before you spend your time doing something that may not be needed. What's the deadline for submitting an SAE report to SWOG? The deadline is always going to be found within the protocol SAE reporting tables. You're going to see different deadlines based on the emails you get from CTEPers. There's a deadline in RAVE. The only deadline that you really need to pay attention to is going to be the protocol. If the protocol tells you to report it within 10 days of finding out about it, you've got 10 days. If it tells you 24 hours, you've got 24 hours. So please ignore the dates that are going to be sent with those automated CTEPers emails that you get, because CTEPers assumes that the day you entered the SAE is the day that it started. And that's often not the case. You often find out about an SAE and it's 6 days later that you find out and report it. We do make every effort to let you know if you've got a report that started, but you haven't quite finished it, you haven't submitted it to SWOG, just let us know that you're still working on it. We'll email you and say, hey, this is going to be due on Sunday. I'm pretty sure you're not working on Sunday. Do you want to submit it today so we can get it onto NCI? So we'll email you with a reminder if we notice that a report's been hanging out in the pending status for a long time, or if it's about to be deleted automatically by the system. RAVE is recommending an SAE report, but the recommendation doesn't match the protocol. Should an SAE report be submitted based on the recommendations in RAVE? The answer is maybe. You always want to follow the protocol guidelines. And if there's any doubt, or if you just like confirmation that, yeah, I am reading this table correctly, just email us. That's what we're here for. We do that all day long. We're happy to help you at any time point. What if a patient's on a treatment arm that has both investigational and a commercial drug? Which table do I use? Do I use both somehow, or do I use one or the other? The answer is that if they're on a combination arm, an arm with a investigational and a commercial agent, the entire combination is going to be considered investigational. So you would only use the investigational SAE table. AEs were submitted in RAVE, then we ran the expedited reporting evaluation. I thought that one of the AEs should be reported as an SAE per protocol, but the CTEPers rules engine did not recommend a report. Is any further action required? The best practice is to use the recommendations in RAVE as a reminder to check the protocol, reminder to email us and confirm. Please don't go by all of the recommendations in RAVE because they are very often incorrect since they're based on such basic rules. On the CTEP-AIRS homepage, there is a help button on the right. It's a little question mark and it says help. There's a lot of great resources there. It's a lot of information, but if you are curious about the system, CTEP-AIRS, the RAVE integration, you can find some help resources there. There's also a lot of resources linked here for information on CTEP-AIRS application, the RAVE integration. Really, the most important thing is just contact us. We would much prefer you contact us than worry about something or stay late or just submitting something that you're not really sure how to do it or what you're doing. If you've never done it before, it's a lengthy, complex process. Call us, email us, we will walk you through that. And that is SAEs in a nutshell. So if anybody has any questions, email us at adr@swog.org. Otherwise, we do have a break next until 10:40. Thank you.
Okay, I hope you guys enjoyed that second break. We're going to continue on for our last set of presentations this morning. I'd like to go ahead and introduce Laura Gonzalez from our operations office in San Antonio. She's going to go over the QA program and auditing.
Hey, y'all. My name is Laura Gonzalez, as Rodney said, and I am the quality assurance manager for the operations office in San Antonio, although I live in St. Paul, Minnesota, so I'm a remote worker. So I'm going to talk to you about the audit program. So the purpose of the audit program, as you can see, is to verify the study data that could affect the interpretation of the primary study endpoints. We check compliance to the protocol and the regulatory requirements and the accuracy of the submitted data. We assess the trial-related activities and the document adherence for good clinical practice. And we also provide educational support for data quality and data management process practices. Many of y'all may have heard, or if you're brand new, you may not. What we say is when we come to audit, we come in a spirit of love and education. So we're not here to come and pick y'all apart. We're not here to point fingers. Everyone on the audit team, all of the nurse auditors, we've all been in your scrubs and your Danskos. We know what you do on a daily basis. We know what the frustrations are. We know what the successes are. We know what it's like to put patients on study. We know what it's like to infuse the drug in the infusion room. So we know what you do on a daily basis. So we're here to help. Okay, we're here to look at what's going on. We're here to see what works, what doesn't work, and how can we help. We're here to provide education and support. So how do we schedule audits? Okay, for the new LAPs, the new members, the new NCORs, we look at new patient registrations and we schedule 18 months out. If you're an affiliate or a component, you'll be scheduled at your next parent institution audit. Institutions that were audited at least once every 3 years, but you can remain at risk for more frequent audits. And then for the FDA registration studies, that's a more frequent monitoring process, and that's in the protocol. If you look way back in section 18, it'll tell you the protocol monitoring process. So for the FDA registration study site visits, the lung map protocol, the initial audit is at 3 months after the first registration to the sub-study, and then every 6 months based on your accrual. 1418, not so much, it's 1806, 1914, the initial audit is at 6 to 9 months after the first registration, and then additional visits are dependent on accrual. And then the new S2302, the Pragmatica study, it's going to be audited on the every 3-year schedule. So it's going to be just like regular treatment audits. So on-site versus off-site, if it's a LAPS, a main member, or an NCOR, We do it on site. If it's a component or affiliate with a large accrual, we'll do it on site. The FDA registration studies are typically offsite or remote, but we can come to your site if you want. So typically when the nurse auditor contacts you, we'll ask. We can do it remote, but if you want us to come on site, we're happy to do it. The in-core components and the main member affiliates are usually audited offsite with the parent institution. And like I said, the FDA registration studies are typically done offsite. So how do we let you know? We schedule them 3 to 4 months prior to the audit. We'll contact you and we'll say, hey, we need to do you. It's your 3-year time. So what's going to look good? And, summertime's coming up. We know it's hard to get those scheduled. We've probably already contacted you. If it's your 3-year time, we want to get that settled up. We send the formal notification about 4 to 6 weeks prior to the audit. So that's when you get your case list and all the instructions that say, here's what you need to do. And we send you a site questionnaire. We send the case list. We'll tell you this is what we need and when we need it. So all the instructions are detailed. And as always, if you have questions, reach out to us. We're happy to help and let you know. So what does the audit team look like? It's a QA representative that can be a non-nurse auditor who's usually the lead. It'll be one or more nurse or CRA auditors. And then it can occasionally be an NCI observer that will come with us. That doesn't happen very often, but they're always invited to come along if they want to come with us. Now, who comes from the site? We want everybody to be part of the audit. Everybody's invited. Okay, so the CRAs, the research nurses, the investigator or the designate, somebody from regulatory, somebody from pharmacy. Like I said, everybody comes. We want everybody to be part of the team. Nothing that we do is a secret. We're completely transparent. We want everybody to be part, everybody to be educated. So what is the audit process? We look at 3 big pieces. We look at regulatory review. So we're going to look at IRB. We're going to look at the consent form content, the delegation of task log, if you have one, or the site authority log, if your protocols aren't covered by DTL. We're going to look at investigational drug accountability. So we're going to look at the DARFs. We're going to come visit your pharmacy, and then we're going to look at patient case review. So that's everything that had to do with your patient, the care. So your chart, if you have a shadow chart, or we're going to look in the EMR. So for regulatory, we look at the IRB. So we're going to look at the regulatory documents for all the protocols on the case list. And that'll be that we typically will only look at protocols that have patients registered. We're not going to look at a protocol that has no patient registration. We're going to look at informed consent content. We do a minimum of 4 consents. We're going to look at your DTL and your site authority log and a trial master file if it's any FDA registration studies. Okay, for local IRB, if you still have any protocols opened under local IRB, we look at your approvals, the initial and continuing reviews and protocol updates, reportable external safety reports and internal SAEs. We need all versions of your IRB approved consent forms or a comprehensive list. We need SOPs for alternative procedures. So like if you have an SOP that you only submit unanticipated events, something like that. So anything you've got going on there. Mostly now protocols are CIRB. So we're going to take a look at your documentation that your CIRB is the IRB of record. So that would be your study specific worksheet approval. We need your approved boilerplate language for your ICFs. We need documentation of your local implementation of protocol updates and consent versions. We need submission of any unanticipated events. So those are your reported local SAEs. We don't need any copies of any CIRB approval documents. Okay, you don't need to save that for us. If it's something that's required by your site, that's fine. You can put it in your folder, wherever that is. We don't need it. We can Google CTSU just like y'all can. So you don't have to provide that to us. So consent form content, we're going to compare it to the model consent. It has to contain all the elements of the required federal regulations. It has to have updated protocol modifications, the specimen and banking specimen banking and the optional studies have to be the same. The questions have to be the same as what's on the model consent. You can't change those. And for the CRB sites, it has to have, if any of your boilerplate language is merged, it has to be approved by the CRB. Okay, so that's why we have to see what your boilerplate is. And if it's been updated since your initial one, we have to see what that looks like. So we can check that when we're doing our consent review. So your delegation of task log, the DTL, if that's for the studies that have been approved, the registration studies, the lung map sub-studies, all of the new studies since August of 2020 for the phase 3, October of 2020 for phase 1 and 2, we'll have a DTL on CTSU. We're going to check that. If you don't have a DTL for your protocol, then you should have a site authority log. And that will cover all of those protocols that do not have a DTL. So we're going to look at that as well. So you don't have to have one for each protocol. If you have, protocols that aren't covered by a DTL, we say you can have one site authority log to cover all of those. And we're happy with that. We'll take a look. For the ones that have DTLs, we're going to spot check those. We review them on CTSU prior to the audit, and then we'll just spot check them while we're there for the audit. So for your trial master file for the FDA studies, it should have the protocol, should have all your regulatory documents, should have your CLIA certificates and your lists of normal lab values and ranges, list of your local SOPs, your site training documents, GCP, protocol specific, et cetera, and a placeholder for any of your centrally filed documents. So all that stuff that's on CTSU, the CVs, the 1572s. And again, you can have this, you can have notes. So if like you don't have your CLIA certificates, they live at your lab, you have a note to file that says our CLIA certificates live at, whatever lab you use. Your lab values, again, you don't have to print out every lab value. You can have a note to file that says we rely on the lab values that are printed on our lab reports, something like that. So you can talk about that when your auditor comes, and we can give you some guidelines, and we have some of that in our FAQs and some of our documents that we provide. So the next thing we're going to look at is pharmacy, investigational drug utility. We review the DARFs, both the IV DARF and the oral DARF that's required for all of your drugs that have investigational, or all of your studies that have investigational agents. We look at the control and the satellite records. We want to make sure that you have complete and timely entries, that you're practicing good documentation, and that patient returns are documented on your oral DARF. Okay, so we're going to look at all of that. We're going to look at shipping receipts, transfer and return forms. We want to make sure that unused or expired drug is returned or destroyed within 90 days of end of use, and no substitution of commercial drug for investigational agent. Okay. And then we're going to cross-reference the DARFs against the protocol records to verify dose and dates of dispensing. We do all of that before. We want to make sure you have an SOP for authorized prescriptions. Make sure that the ordering investigator has to have an active CTEP account. We do for on-site audits, we're going to do a tour of the pharmacy to assess security, storage conditions, verify the inventory. If we're doing an off-site audit, we can do that tour via FaceTime, WebEx, Zoom, any of those kind of platforms. We can set that up. And we usually do that, when we're making the scheduling with that, we can talk to your pharmacy folks and say, whatever time is good for that. And we try to be very sensitive to the workflow at your site. So we know if y'all are super busy in the morning, getting everything ready, then we don't want to get in the middle of all of that. And we will work with you and your team to set that up when we're doing the scheduling. So now we're going to look at patient case review. And when we set up our case list, we look at 10% of your SWOG and CTSU accrual. We try to get 10% of treatment and cancer control cases. We take a minimum of one case for each of the non-SWOG FDA registration studies. We do a minimum of 3 cases when we're looking at that big reg list. And then when we come on site, we're going to do one unannounced case. So you won't find out about that until the day we show up. And when we do an unannounced case, we only look at informed consent, eligibility, and data quality. So it's kind of like a mini review of the case. So when we look at patient case, we look at informed consent, we look at eligibility, we look at treatment administration, disease and endpoint assessment, toxicity assessment, and general data quality. So those are the categories. So for chart preparation, a shadow chart is acceptable for us. We're fine with that. We recommend that when you organize your chart, that you do consent and screening and eligibility, and then chronological by cycle and reporting period. So H&P, labs, disease assessment, et cetera. So if you're setting up a shadow chart, that's the way we like it. I'm an old nurse. I like a shadow chart. Some of the newer nurses, they like the EMR. That's fine. However y'all do it, we're cool. We can work in any kind of a situation. If you're doing a shadow chart, we provide instructions on how to flag by color coding. We want the specimen submission documents flagged. You got to print out the specimen tracking documents so that we can see that you've submitted the specimens appropriately. And if we're going to be reviewing in the EMR, we have an EMR source documentation locator form that we'll send you. So we'd like for you to fill that out so that we know where we need to go in your EMR. We're pretty good with Epic and some of the other Meditech. Some of the ones, Epic is like the biggest one that we work in, but we can pretty much figure out where we need to go when we're working in your EMR. Okay, so for informed consent, what are we looking at? We need to make sure that the most current version is signed prior to registration, contains all the required signatures. If patients need to be informed of new findings, that it's done in a timely manner, that the specimen banking and optional studies are offered and the intent reported correctly in open at the time of registration, and that a HIPAA authorization has been signed. For eligibility, we're going to verify diagnosis by reviewing pathology or any other diagnostic reports. We're going to review the medical history for any exclusion criteria. We're going to verify that all the pre-study assessments to meet protocol requirements were done and were performed within the specified time limits. We need to make sure that the eligibility affirmation is signed and there are no exceptions granted. Okay, NCI says no waivers are allowed. Study chair, you can't call up the study chair and say, yeah, we got the labs a day out of window. Is that okay? Study chair will say, yeah, that's fine. And you've got a note to file that says the study chair, it was okay. Said it's okay. We'll say, yeah, sorry, study chair can't say it's okay. Not eligible. Sorry. So just make sure that you're going through your eligibility criteria. For SWOG protocols, that's section 5. Go through every one. We always recommend have second set of eyes looking at it. Make sure that they meet all the criteria for eligibility, okay? So treatment administration, we're going to look at dose calculations, BSA, was everything calculated correctly? We want to verify both the drug orders and the drug administration. So we need to see your treatment plan. We need to see the chemo orders. Like I said, I'm an old nurse. Back in the day when the doc used to write the chemo orders and sign them off, that's what we want to see. If it's a treatment plan in Epic and Beacon, then we want to look at that too. We need to see that dose mods were appropriate. If we had to change because of a AE or because of weight change or whatever that was. If they have patient diaries, we need to see those. If the patient forgot to bring their calendar back, we know that happens. So then we just need to see a note in the chart that says Mrs. Gonzalez forgot to bring her calendar, but she told me she took all her medications and she has her pill bottle and it's empty. So we're verifying 100% compliance. That's fine. We understand patients lose their calendars. They forget to bring their drugs back. But as long as you can document that you had a conversation and that the patient says they took all their meds, we're good with that. And then if you've got any documentation to support delays, deviations in treatment, whatever those are, write it down. Anything that goes sideways in your protocol, write it down. Anytime patients are scheduling issues come up, they don't show up for an appointment, the insurance won't pay for their scan, anything that, anything that goes sideways, write it down. Document, document, document. So then endpoint assessments, we're going to look at make sure the disease assessments were performed per protocol. Did they get their scans done on time? We're going to review those reports, path reports, radiology, lab, physical examinations, all of those. Make sure that the same method of measuring the disease assessment that was done at baseline and at each assessment, tumor measurements are documented and recorded. Your RECIST log needs to be part of your chart. Off-treatment follow-ups are conducted per protocol. Make sure if a patient doesn't start treatment, make sure you check to see what the protocol says about how they need to be followed, even if they don't start treatment. If they come off treatment early, make sure that they're followed per protocol. Make sure you understand what your requirements are for that. Adverse event assessments, make sure we want to make sure that the baseline and the follow-up studies are performed. Grading and attribution of AEs are documented and signed off by the investigator or the qualified practitioner. Make sure we recommend an AE log. We love AE logs. So if your site doesn't have that, you might want to think about how y'all are documenting your AEs and that they're being documented appropriately. And then also for the protocols that have immunotherapy, are we documenting is the immune-related status, if that's applicable? We want to make sure AEs are reported appropriately and that SAEs are reported in a timely manner. My colleague Maggie was just talking to y'all about SAEs, so if you have any questions, Maggie is the guru for SAEs. I can help out with some of the AEs if you have questions about that, but Maggie's your girl for SAEs. And then general data quality, adequate source documentation. Anything that you enter into RAVE, you got to have something in your hot little hand that supports that entry. So just remember that. If it goes in RAVE, there needs to be a piece, I'm saying a piece of paper, because again, I'm an old nurse. There needs to be something in your EMR or something in your chart that supports that entry. So if you don't take anything else away from this, no, whatever goes into RAVE, you got to have something to support that as source documentation. And especially for some of those like cancer control, where they put in a lot of QOLs and they ask a lot of really weird questions. If you want to use a CRF as your source document, that's fine. We're happy with that. Just make sure it's signed and dated. Anytime you use something that's not like part of the medical record, part of the chart, that's not an office visit or a lab report or something like that. If it's something a little weird, because I know sometimes we ask some of those, those weird QOLs that we give or those weird assessments that we put in, that's fine. If you want to use that because it's not something easily abstracted from the chart, you can use a CRF as a source document, but it needs to be signed and dated. So we just want to make sure that all of that data is accurately reported, timely submission of data, specimens, images, questionnaires are submitted per protocol, good documentation practices, sign it, date it. If you make a mistake, line through it, initial it, date it. So the exit interview is going to be at the end of the audit. We will have already reviewed each case with the appropriate nurse or the appropriate CRA. We will have gone over all of our findings. When we meet at the exit interview with the PI and the staff, we typically will only review the major deficiencies and summarize our findings. We'll answer any questions. We'll submit a preliminary report within 24 hours of the end of the audit, and that will just list only the major deficiencies. So if we have, at the end of the audit, we'll have 3 audit ratings. If it's acceptable, there's no major deficiencies, we'll see you in 3 years. If it's acceptable with follow-up needed, there will have been some major deficiencies, but nothing really great big. So we need a written response, including a corrective and preventive action plan called a CAPA that you have to submit. If it's unacceptable, that means that you had enough major deficiencies that we're a little bit concerned. So we want to come back and see you probably in about 6 to 12 months. So we need a CAPA. If you've been unacceptable more than once, We're going to ask for the CAPA, we're going to see you in a year, and we're going to ask for a site improvement plan. And so that's a little bit more involved than just the CAPA. There's probably some process issues or some structural issues at your site that we need you to think about and see if you can work on. If it happens usually more than twice, then there could be a chance that your registration privileges are going to be suspended. I've not seen that happen yet, but there's always that possibility. So helpful hints: take lots of notes, sign them and date them. Never, 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 never use whiteout. Keep your records on a real-time basis. Document your height and weight and performance status. So train your docs. We need the performance status. Keep your logs for tracking adverse events. Like I said, we love AE logs. We love ConMed logs. So concomitant medications, keep those in a log too. Those are easy because a lot of the studies want ConMeds. If you keep it in a log with a start date, a stop date, the dosage, how often they take it, that's easy for us to track and easy for us to go back and audit. Like I said, 2 eyes on that eligibility criteria is a great thing to have. Look at an audit as a positive learning experience. Like I said at the beginning, we come in a spirit of love and education. We're not here to pick you to death. We want to help you. We want to provide education. We want to be supportive. Always include, if you're a big NCOR or a LAPS or a main member, include your affiliate and your component staff in the audit process. Try to conduct internal audits and training so that you can be ready for when we come. Look at the CRA workbench. There's lots of reports on that. You can use those to help you. So if you have your IPR and expectation reports, if you have outstanding queries, if you have the list of ineligible patients, those are some of the reports that you can use. Here's some additional resources. I'm running out of time. So you can use those to help you get ready. Again, some more links and additional resources that are on the SWOG website. Does anybody have any questions? QAmail@swog.org if you have any questions. If you want a copy of this presentation, QAmail@swog.org. I'm happy to send it to you. Like I said, we are completely transparent. We want to be helpful. We want to be supportive. We don't want y'all to be scared when we come to your site or when you see that email that says it's time for your audit. We want everybody to say, oh, goody, the auditors are coming. Okay, so I want to thank you for your time. I want to welcome y'all to SWOG. For those of y'all that are new, I want to tell you how happy we are to have you. And I hope you love, love, love working for your new site, and you love, love, love working with the SWOG audit team. So thank you.
Good morning, everyone. My name is Laura Wells. I'm a data coordinator at the SWOG Statistics and Data Management Center in Seattle. I'm going to provide an overview of SpecTrack, show you how to use the system, and then let you know where you can find more information. Hopefully this will answer any questions you have, but if not, you can ask questions at the end of the presentation. So what is it? The specimen tracking system, often referred to as SpecTrack in conversation and as the STS in writing. It's a web-based program that allows you to log specimens, keep track of shipments that are sent to the SWOG laboratories and repositories. When you complete a log line in specimen tracking, it resolves the expectation for that specimen immediately, and it allows you to track when a specific shipment left your institution and when the lab receives it. Okay, so it was first released in November of 2003. S221, a large phase 3 adjuvant breast trial, was the first study to require its use. It's been 18 years since then, and now all of our studies currently use STS. So most of us here today haven't actually used the original version. I haven't. And as is the case with most new systems, there were a lot of kinks our team had to work out. And we want to stress this because some of those changes and modifications were initiated from comments from the sites. So we really take constructive criticism into consideration and hope that we can make changes and make everyone's lives a bit easier. Okay. So how do you know if your study uses the specimen tracking system? The best way to check is by reviewing section 15 of the protocol. This is the special instructions section, and it'll provide all the details required for specimen submission, including collection, processing, and special shipping instructions. Another way to know is through the patient consent. If specimens are collected for the study, the consent will indicate that. It'll also let you know if specimen submission is mandatory or optional. And then lastly is section 12, which is a rare section for protocols that are not FDA registration trials. But if the protocol has this section, then it'll tell you when and how to submit those specimens. So listed here are some of the best sources of information for using the system. As mentioned on the previous slide, section 15 of the protocol will tell you what specimens are required or optional. There are also, if your question is only about using the system, there's a downloadable tutorial on the SPECTRAC homepage. And then there's also a link to a training module that you can watch and interact with in order to learn the system. But if you've used these resources and are still having trouble, you can reach out to your data coordinators. We can't solve technical issues, but we can help with general information on the requirements and protocol clarifications. And we can also walk you through the system using our test system, which is what I'm going to go through next. So this is the CRA workbench. You can access the CRA workbench directly from SWOG.org or through CTSU. SPECTRAC is one of the links you'll see on the left. So from here, click the SPECTRAC link. And then it'll bring you to the sign-in page. On the top right corner, you can see that I'm going to be using the test database. To get started, you'll need to enter your username and password, and those are going to be the same as your CTEP credentials used to log into the CRA workbench and open and other SWOG sites. So once this is done, just hit sign in. And then you may see a list of institutions like this, where you'll need to select the site responsible for the patient. And you want to make sure that site matches what is entered in open for the patient. And if you're only associated with one site, you won't get this drop down. It'll just automatically take you to the database. So this is the specimen tracking homepage. As you can see very clearly on the top, this is a test site, and this text won't be present for you. And then the two links you're going to use the most are the log a specimen and specimen manager, which we'll be going through those in depth. But then there's also a view and update consent link that you can review the consent questions and change answers if you need to, if there was an error or if the patient changed their mind. And then notify that a specimen can't be submitted is the link you would choose if something can't be submitted for some reason, like it wasn't done. That's how you would notify the lab there. And once you complete that, it also resolves the expectation immediately. And then reports is where you'll find assay results if your study utilizes this feature. And then administration. That's where you can add contact names and email addresses of people who will be automatically notified when a shipment has been received by the lab. So if you have any personnel changes, please make sure you update contact information to ensure the proper staff is notified. And then the last link here is contact us. This will give you links to send a message to our technical team. So it goes to technicalquestion@crab.org. And whenever reaching out to them, please provide as much information as possible. If you have it, patient number and study ID can be very helpful. And this will just get the right person answering your question a little faster. Okay, so when you click log a specimen, you'll go to this page. The first thing you'll notice is no patient ID yet. And this is only for studies that allow pathology or cytology submission prior to registration. So if your study requires this, you can choose this link and enter the demographic information, and then the system automatically assigns you a patient ID. And then if that patient is registered to a SWOG study, they'll continue to use that ID number, but if they're not, then it'll just be deleted. Okay, and then you at the bottom, all fields with an asterisk are required. Okay, so enter the SWOG patient ID number, initials, and study number, then click next. It'll take you to a page that looks like this. One of the most recent changes you'll see right at the top is the filters. I think Phyllis touched on this as well. But you now have filters to narrow down all the specimens you're actually going to be looking at. So if your study only uses one lab, that'll satisfy the requirement immediately. But otherwise, you can choose a lab, specimen type, submission time point, and hit apply to make your list smaller. Using these filters reduces specimens to choose from and should help cut back on errors. Okay, so once you've entered the patient ID, only specimens for this patient will appear. You'll want to click on the blue hyperlink for the one you are trying to submit. There are different columns, registration step, time point. You want to pay close attention to the time points because some of the specimens repeat themselves. So you want to make sure you're not submitting for the wrong time point. And then there are two options for, oops. There are two options for pathology submission here, the blocks and the unstained slides. So on the other column, it'll tell you which one is preferred, and you only have to ship one of these, and then that'll automatically resolve the expectation. Okay. And then you can tell that a specimen here has been successfully logged with the green check mark. And then if it is reported as unsubmittable, there aren't any examples, but it'll have that red X icon. Okay, so this is the final step. There are only two required fields here. Quantity is pre-populated, but you can change it if you need to change it. And then the date is the other required field. The contact information is set up by you, but it is optional. It just helps the lab know who to contact if there are any issues. Otherwise, it'll just contact the name and email of the person who entered this information in the system. Okay, once you've entered your information, it'll take you to this page. It kind of looks like nothing has happened, but at the top you can see the green light shows that it was successfully logged. And there's also the yellow light, so you know that it's not shipped yet. Okay. So there's a few things you can do down here at the bottom. When you choose update specimen, you can go back and edit what was entered. You can also delete the specimen at any point. You can log another specimen for this person. That just takes away the first couple steps of entering the patient ID number. You can also click on home, and that'll take you back to the home page for STS. And then you can hit specimen manager, which is where you'll go to complete the shipping. So that's what we're going to do. Okay. So this is the specimen manager homepage. It combines your specimens and your shipment information onto one page. Specimen manager will show all specimens logged by the institution you choose in the first screen. It's useful how to know how to use the filters on the top of the page there. You can filter by patient number, study number. You can choose if you want to see things that haven't been shipped, have been received. It's also helpful to add the receiving lab filter if you're sending to multiple labs. Okay, so once you've made your selection, your screen will look more like this. It's important to remember that these expectations are not going to be resolved until the specimens are shipped in the system. So as you can see, this one is still showing up as not shipped and has not been resolved. You can also delete from this point as well, in case something was entered incorrectly. And you can put multiple specimens into one shipment by clicking that box. This only has one line item, but you could have more. And then if you are shipping multiple specimens, make sure you include the packing lists in the box. And also make sure that your study allows for batch shipping. Okay, so once you have chosen create a shipment, you'll be taken to this page. Step one is going to be just verifying the information you entered at the top of the page there. If you need to make any changes, you can do so by clicking add or modify shipment contents. Step two is you want to verify the pre-populated lab info and just confirm in your protocol that this is the lab number that it should be going to. And then the comment section is a place to put any important instructions or requests for the lab. For example, if you're sending more or less than what the protocol requires, you would explain why that is in this section. And then step three is the final step here where you enter the date. You can enter a future date for up to 5 days in the future, and you can also backdate it if you've already sent the shipment, but didn't log it, but that's not recommended and should be avoided as much as it can be. But if that does happen, it is possible to backdate them. Okay, after the date is entered, you're going to click ship this shipment and generate a packing list. Okay. So this shows a successful shipment, the shipment number, contents, lab address, and so on. You can generate a shipping label if needed, or return to the specimen manager or the home page. For this example, we're going to click on View Packing List. So this is where the View Packing List link takes you. It opens a new tab and shows you the information that has been entered by you for the shipment. The consent question answers are also at the top there, the shipment number and shipping information. So you'll want to print this packing list and include it with your specimens. You only need to print the packing list and not other screenshots, and you don't need to send this packing list anywhere other than with your shipment. So this has all of the information that you need. It lists where you're shipping to, consent questions again, shipment number, barcode, who the shipment is by, and who you are, who you are shipping it to. Next one. So here's an example of what the shipment manager or specimen manager page might look like. This is a different patient number, so you can see a few more different statuses. So you'll notice that we filtered by patient number, and they have 3 received shipments and 3 shipped shipments. So when the lab does receive a shipment, it'll auto-populate the receive date and condition column. So nearly everything is fixable in this system. It's not possible to ruin anything or break it, so always keep that in mind. This should only take a couple minutes tops. So if it's taking any longer, there might be a problem. And if you ever get stuck, there are resources at your disposal. There's a tutorial. You can email and call for help. We encourage emails to the technical question link. There's multiple people on there, programmers, data coordinators, database administrators, and others. So there's a lot of eyes on that. And the correct person will respond to you. Okay. If there are time restraints, the DC assigned to the study can also offer assistance. And we have the DLs on this page here. So it's best to use the DLs here instead of the specific DC's email. Just in case someone's out, then a data coordinator covering can respond. And that's it. Are there any questions? Perfect. Now Hannah Brown will give us some specimen shipping tips.
Great. Hello, everyone. My name is Hannah Brown, and I'm a biorepository protocol coordinator at the SWOG Biospecimen Bank in Columbus, Ohio. Today, I'll be presenting some tips for submitting specimens to the bank. First, I'd like to give you an idea of who we are at the Biospecimen Bank. We are part of the Biopathology Center at Abigail Wexner Research Institute at Nationwide Children's Hospital. We serve as the biorepository for several major groups and organizations, including SWOG, Children's Oncology Group, NRG Oncology, and NCI Early Phase and Experimental Clinical Trials. On an average day, the Biopathology Center receives 100 to 160 packages, which can contain upwards of 1,000 specimens for all of the groups that we serve. We receive several different specimen types for SWOG protocols, including FFP blocks and slides, fresh blood, bone marrow, stool, and urine, and frozen blood products, urine, and tissue. We accept all specimen types on weekdays, Monday through Friday, and we accept shipments of fresh blood and bone marrow on Saturday for immediate processing. Accurate specimen submission is crucial to our day-to-day operations, and double-checking your specimen collection, specimen labeling, and paperwork prior to submitting can help make sure that things run smoothly. When reviewing the SWOG protocols for specimen collection guidelines, protocol sections that are particularly helpful include Section 9, the study calendar, Section 15, special instructions. The study calendar includes general information about specimen collection time points, and Section 15 provides more detailed instructions regarding submission requirements, labeling, processing, and shipment. Section 12, discipline review, also covers whether the protocol includes quality control, pathology review, or central review of specimens. Biospecimen processing and submission procedures, when not provided explicitly in the protocol, can be located on the Biospecimen Resources tab on the SWOG website. If instructions are provided in the protocol, please use those instructions instead. Other helpful resources can be found on this page, including instructions for labeling and labeling templates and shipping addresses for labs 200 and 201. That's us. In 2022, we had over 8,000 SWOG specimens arrive with labeling issues. The labeling issues for over 1,000 of them may have delayed their processing. This is one of the most common mistakes we see across all of the groups that use the Biopathology Center services. As a quick overview, most specimens are required to be labeled with the SWOG patient ID number, patient initials, date of specimen collection, and specimen type. Additional labeling for FFP materials include tissue type, surgical pathology ID or accession number, and block number. We are unable to assume any of the required labeling information, and missing information will result in the bank contacting the submitting institution for follow-up. Some submission issues may result in a query. Please note that the S2101 IMatch protocol has special labeling requirements, including the SWOG specimen number. If your site is involved in the study, please review the current version of the protocol for the most up-to-date labeling requirements, or reach out to us if you have any questions. As I mentioned previously, there are some convenient labeling templates on the biospecimen processing and submission procedure page on the SWOG website, which can be very helpful in making sure that all the labeling requirements are met for each submission. So once your specimens are collected and you have made sure that they're appropriately labeled, you have your packing list, it'll be time to prepare the shipment. Make sure that you have the required paperwork, including that packing list and the redacted pathology report for any FFPE specimens. Make sure that the information across all forms is correct and matches your specimen labeling, especially collection dates, quantities, surgical pathology ID, and block number. When redacting the pathology report, please remove patient information that is not required, such as patient social security number, medical record number, account numbers, and full face photographs. Make sure that you leave the SPID, block number, collection date, diagnosis, results, and any other information about the specimen visible. Please also make sure every page of all paperwork submitted is labeled with a SWOG patient ID number. We ask that you only ship frozen specimens Monday through Thursday to arrive Tuesday through Friday. On Saturdays, we only accept ambient blood and bone marrow specimens. We have people frequently ask about batch shipments. We can accept small batches unless otherwise stated in the protocol of no more than 5 patients or 50 frozen vials or 200 slides, whichever is fewer. Small batch sizes help our labs process all the specimens they receive in a timely manner and avoid large loss of specimens if something were to happen to the package in transit. Please place each patient's specimens in a separate labeled bag, appropriately labeled to make sure that they don't get mixed up. When you are packaging your shipment, please keep in mind the weather outside. When shipping frozens, always include plenty of dry ice to prevent thawing regardless of the weather. For ambient shipments, include a refrigerated, not frozen, cold pack unless otherwise stated in the protocol or kit instructions. For example, a lot of our protocols use Trek tubes, which should be submitted without a cold pack as it can damage the specimen. During colder months, make sure that you insulate well with bubble wrap to prevent specimens from freezing. Our preferred shipment method is FedEx Priority overnight. Other carriers and methods may delay receipt. Remember that when shipping, even if the weather is fine in your city, the specimens are traveling from your city to the FedEx hub to the SWOG bank in Columbus, Ohio, so it may vary. Some common specimen quality issues that we see at the bank include specimens arriving without sufficient dry ice or even thawed. To correct this, make sure you're choosing a large enough container to hold enough dry ice and that you are sandwiching the specimens in the dry ice so that they remain completely covered during transit. Sometimes blood or bone marrow specimens may arrive hemolyzed or clotted. To avoid this, ensure you mix the specimen in the tube immediately after collection by gently inverting 8 to 10 times. To avoid leaking specimens, make sure to use plastic tubes for frozen specimens and not to overfill cryo vials. After packaging a box of slides, gently shake it. If you hear rattling, the slides are more likely to break in the box as they're not adequately cushioned. If you ever have any questions about what type of specimen to collect or time points, you can refer to the protocol or contact us at the bank, as we are always happy to assist. Some common shipment issues that we see include missing paperwork, missing labeling information, and discrepant information between the documents and the specimen labels. Double checking the paperwork and the specimen labeling prior to sending the shipment can help to avoid some of these errors. Also keep in mind that dry ice sublimates at a rate of 5 to 10 pounds every 24 hours. So including at least 5 pounds of dry ice in your shipment can help prevent your specimens from thawing during transit. I've included a few helpful sites for everyone. The previously mentioned biospecimen processing and submission procedures page, and the BPC kit management page for ordering biospecimen collection kits when they are provided by the protocol. And finally, I'd like to provide you all with our contact information so you can reach out to us if you ever have any questions about specimen submission. If you would like any of these notes or slides, you can also reach out to us there. Listed here is our phone number, email, and address for the solid tissue and leukemia divisions of the Biopathology Center. You can also find our address on the packing list when you prep the shipment so that you know that you're sending it to the right place. When corresponding with our teams via email, we ask that you reply all when responding so that our team can better assist you. Great. Thank you all for your time. Does anyone have any questions? Okay, thank you.
Well, just wanted to thank everyone for sticking around for the last talk. So the standard joke at this point in time is the fact that I'm the only thing that stands between you and lunch. And so while we're
just last one.
And it's been a long-standing SWOG tradition that they always put the statistician right between any presentation and lunch. So you can hear me okay? I have way too many slides. So for those of you taking statistics courses, this always happens, right? There's way too many slides and they have too many formulas on them and you really don't care what the guy's talking about. And so I'm going to try to fulfill that role. My name is Mike LeBlanc. I'm the director of the Statistics and Data Management Center. And I know it's kind of obvious to say your scientific role, but I really want to, it's always good to say these things that everybody knows anyway. So this so they're ingrained in your head that as statisticians, as clinicians, and you look using the results of our studies, none of it works without high quality data, high quality interactions that you might have with other staff entering the data, with the patients. Getting that data in is really critical to make everything work. And as I said, I have too many slides, but I wanted to start off with a huge positive result coming out of SWOG in the last year. And it was for high-risk melanoma, and it was neoadjuvant versus adjuvant chemotherapy. And how many of you can actually, if I only get through 3 slides, I will get through more. How many can actually know what these 2 curves represent? Okay, some of you. So these are Kaplan-Meier event-free survival curves. And the way you look at them is actually the neoadjuvant therapy was do I have a pointer here? Is this a pointer? Ooh, I guess that's a pointer. So the neoadjuvant therapy was the new treatment, and this was adjuvant. And you can look at the curves and along the x-axis, if you look out to 18 months here, about 80% of the people had not had an event with the new therapy versus about 50%. You kind of think of proportions, but proportions happening over time. And so it's a standard thing that you'll see in journals. And it's actually critical to the data that's coming in to be actually get good estimates of what these things are. So these curves, because the curves are farther apart, shows that the new therapy works a lot better than the old therapy. And then out here at 36 months, it's about 40% versus 80%, 0.8% without an event versus 0.4%. So it's kind of like running proportions if you're looking at these 2 curves, telling you how well that new therapy is working. But to actually trust those data, we need to get the right data coming in. This is another example from an earlier study in myeloma with a much larger sample size, a randomized phase 3 study. And the blue line, again, was above the red line. And if you look at this curve, say at 24 months, it was about 50% of the people who are if you look out at 24 months, about 50% of the people were surviving with the standard therapy, up here at about 50% at 24 months, versus about 70% with the new therapy. So these curves are the output of all the data that's being collected for a trial. And so I wanted to just go through a little bit of the statistics of the background of how the sort of the design. And then I'm going to hit a couple statistical concepts, and I'll show one formula, but the formula is to make a point, really. So clinical trials are denoted in multiple different phases, phase 1, phase 2, and phase 3. Within SWOG and NCTN group, We typically focus on phase 2 and phase 3. Phase 1, early testing. You may do a lot of those studies on single institution studies that you're a part of. Phase 2 studies typically are looking for promise of treatment efficacy. They might be randomized, might be not randomized. Also looking at more toxicity data. And finally, phase 3 studies are these large randomized studies that really try to prove the new therapy is better or not better than the old therapy, basically, when it comes down to it. And that was that last slide I showed of the curves corresponding to a phase 3 study. And of course, you're collecting, we're evaluating therapeutic interventions in multiple ways. We're looking at both early on in those studies in terms of biologic activity, obviously safety and toxicity, but then also clinical efficacy, and that might be clinical response. It might be patient-reported outcomes, recurrence, progression, survival, all the things you're familiar with actually, and your staff actually collecting data on these aspects of it. And then other long-term data can be important for certain settings as well. So now the statistics part, there's 2 components I'll talk about, 2 terms you've no doubt heard multiple times, variability and bias, and talk about all of our roles in dealing with these 2 components. And what problems they cause in the context of clinical trials data, and how can they be prevented or reduced somewhat. So here's a graphic I took from someone's course, and I probably didn't, I attributed in very small print, but the concept of variability and bias, thinking of arrows against a bullseye. If there's something that has a low variability and low bias, it means all those observations, all those arrows are all at the target, the thing we're really interested in. That's low variance, a low bias. If it's low bias, you're getting to the target, but you're kind of like me, they're all over the place, right? They're a lot more noise around it. And if something is both inaccurate, or in other words, it's biased, it's not taking you to the true thing you care about, but it's still precise. That's that third diagram where all the arrows are going to one spot, but they're off. That can happen sometimes in trials if they're not performed correctly. And then, of course, there's inaccurate and imprecise, or high variance, high bias, and the arrows are going all over the place and they're off the target. That's probably how I would probably be. For that, I was being too generous to myself. I actually know where I was shooting. I'd probably be off the side. But we're trying to control those concepts, but in the context of clinical trials. So how do we control variability? That's one of the things that every one of those protocols you see that has eligibility criteria. We're interested in patients that of a specific attribute. So for instance, a really basic one might be patients with local disease and low performance status might be eligible for a specific trial. We're doing that for a couple of reasons, actually. We're doing it for safety, but we're also trying to get a focused group of patients. We're kind of controlling bias by that idea. Another way we control bias, and it's a standard statistical thing to control bias, is with collecting data on more patients. I mean, variability is collecting patients with more data. So a larger trial, the last thing you want to do is collect data on 100 patients when you could have collected on 5. But we're doing that statistically so we can get more precise estimates, so we can figure out whether that treatment really works, whether there's really toxicities with respect to that treatment. So that increase in sample sizes for a real reason going on in a clinical trial. And what is your role in doing your role? Actually, all of our roles is making sure those eligibility criteria are actually applied to. And other big ones, avoidance is really following the protocol, avoiding deviations where all possible, both in terms of treatment as well as follow-up information. I'll give an example in that in a bit. And then, as I said, not just that the assessment timing, but also the timeliness of data submission. That's another way we can control variability. Bias, that other statistical concept, is that it's a tendency for a tendency for the statistical result to differ on average from the true state of affairs. So that was that bullseye where all those, think about those arrows are all going off to the side. We really wanted that one target was different. And that can happen because of flaws in clinical trials, which is really bad. It's the sense that we think we want to estimate the true difference in the proportion of people who are alive and doing well at, say, 3 years. But for some reason in our data collection systems, we're getting something completely different. We have to try to avoid that. So for instance, if a study intended for treatments with local disease only, but includes a number of patients with more advanced disease, and we're just trying to estimate what the outcomes are, that's going to be a biased answer if we're really interested in local patients. So if we include patients who really are not the ones we want to draw conclusions from, that can introduce bias in terms of our interpretation of the results. And so again, a solution is make sure we're adhering to eligibility criteria. This is an example I'll follow up with yet a little bit more detail. If for instance, patients are followed differently on one arm versus the other, that can introduce bias depending on how those patients are followed. And that's a big deal. We try to keep track of that really closely, but that's really very much in your hands when you're looking at that, as well as the design of the study. So here's an example of illustration of the impact of loss of follow-up. And so this is a hypothetical patient called patient A, a registration date of March the 1st, a first assessment date of May the 1st, a second assessment date of July the 1st, and a progression date in October the 1st. And that one patient is on treatment 1. Now we suppose we have another patient, patient B, on treatment 2. And I should have pointed out that patient A was actually saw to progress on October the 1st as well. So that's what I forgot to mention, that purple arrow at the end. Patient B registered on March the 2nd, assessment date May the 4th, so pretty tight timing of follow-up. And their assessment date, their third assessment date is July the 1st, but they progressed. That's that purple area there. So we have one progression on October the 1st for a patient in treatment 1, progression at July the 1st on patient in treatment 2. As I said down there in the words. So this is not, now let's take this, let's think about the same data. So let's just go back to patient A again. Registration date, assessment date, progression date, October the 1st. But on patient B, on treatment 2, let's suppose that patient was lost to follow-up on April the 1st. So we never got to see assessment date 1, assessment date on May the 1st, or assessment date on July the 1st. And includes, we also missed the progression. So what would happen is now the data set that I'm using as a statistician to create those curves is based on inaccurate data. And we're going to analyze data that says there was a progression for patient 1 on treatment 1 on October the 1st, but no progression on patient B. Patient B is doing fine as far as we know it. And that's just because of that lack of loss to follow-up. That's one of the reasons why you're always pushed really hard to make sure you follow up per protocol on patients. Because that loss to follow-up, if those data are missing, we're going to draw the wrong conclusions. So bias, it's first off, it's designing the protocol, writing the protocol appropriate, but it's adherence to what the protocol requirements are for follow-up evaluations where at all possible. And we don't, there's reality on the ground. So it's where all possible. It's schedule the patient's return to evaluations for the protocol schedule, absolutely critical. And then they have the tests performed per protocol, because some of those tests are clearing the protocol for both safety as well as efficacy reasons. And your role or your staff's role is making sure that in terms of doing that, it's verification of eligibility, it's adherence to protocol guidelines. And so basically variability in survival data here is time until event. Basically it's time until something happens. It's almost often the most important event in a study. For all the data we collect, most of those phase 2 trials are based on that time until event. That's why I'm emphasizing it so much, and it's so critical, your role in that. And incomplete data increases, as I said, both the variability and how long until you see that event, as well as potentially induces bias between the curves. So I just gave 2 examples. I'm just going to give 2 curve examples. It gives me a chance to describe survival curves again. statistically, but also it goes back to those two hypothetical patients. So here's a pretend study that was designed, it accrued over 3 years. It had 1 year of follow-up. So it means patients came on study for about 3 years, and we followed a patient's additional 1 year past that point in time. And these are those same Kaplan-Meier curves that I showed before, where the experimental treatment is the blue and the experimental actually is the yellow, yellowish brown, and the standard is the blue. So at the end of this study, these patients looked about the same, and the x-axis is survival probability. So if you went out to 1 year, about 50% of the people are surviving on both of those treatments. They're basically the same. And then the bottom bar just shows the last contact date for patients who we lost to follow-up or that we don't have the data for yet. However, for the same reasons that I showed patient A and patient B, if in fact we lost some patients to follow up on one of those arms, this is the result we would have seen. So this is the same data. I created this data in my statistical lab. I created those data, so I had complete control over it. And now I said, okay, what if we lost some of the patients on one of the arms, sort of preferentially patients, especially patients, what if we lost patients who had a tendency to live longer anyway? What if we lost some of those? Then you would incorrectly see that experimental therapy is better than the standard therapy. There's separation in those curves. That's what that yellow bar above the blue bar means. So we'd end up with a wrong answer just because of differential patient follow-up. So that's taking that one patient on one arm and one patient on the other arm to taking to several patients. And this is the impact of the study. So we draw entirely the wrong conclusions. We might conclude this, for instance, this therapy is worthy of a larger study that try to see it works. We might start treating patients inappropriately. So it has real implications in terms of everyone because of loss of follow-up that happens. So what do we need? Complete and timely submission of accurate data and thorough documentation of all eligibility criteria. And then complete description of patient treatment received, and evaluation of objective status and toxicities, obviously, at those evaluations as well. So this is my one formula. So and it's a special formula, and I take really it's not the numbers per se, but it's so if you had a sample size and you had some non-adherence rate, what would be the sample size to get you the same precise estimate with some non-adherence? And we're going to make the best case scenario here that non-adherence is not biased non-adherence. So you think that, I mean, I would think like if you lost 10% of the patients, it means you needed 10% more patients on that study. But in fact, it's not 10% more. It goes like the square. So it's the sample size divided by 1. So for instance, if I had a 100-patient study, and take that top row, 100 patient study, and I lost 10% of them due to a non-adherence rate, to get the same precise estimate, the same conclusion, I would need 123 patients. If I had 20% of the patients non-adherent, it would take 156% of the patients. So it's this thing we kind of, we're thinking about comparisons here, and it works out that if you have incorrect data on one of those two arms, it actually has a squaring effect in terms of how bad it is in terms of the strength you can draw conclusions. So the moral seems high-handed, but okay. So the moral of the story is that if you get data on, if the data is not submitted on one patient or one patient's not adherent to the protocol, at a minimum, it's like not valuing the data for a patient you've never seen. So every patient's data are valuable, they're valuable, but we're saying, in fact, that not only are you ruining the data for that one patient, you're ruining it for another patient who's contributed and you have the data on. So it's a high bar, right? It's a study, even though they're independent individuals, they have to work as a team. We all have to work as a team to get the best data, because that impact is really, really funny. And if it kind of blows your mind, it still does to mine as well, because it's kind of weird that somehow one patient can influence another, but it does in those effects. Okay, so that was the one formula we got through there. So the big bold print, high quality data is essential for good studies, and obviously your efforts, all our efforts are essential for that to happen. And it's not just for the randomized phase 3 studies. It's not just for the FDA registration studies. All of those studies are making conclusions that we have to make to move studies up the line to make a difference for patients. Okay. And why is it always critical? It's because we're looking at the data all the time. But we're not just, it's not this, we do a 5-year study, oh, we'll catch up to it years. I know some of your institutions might get a nasty email note from me saying, oh, you're not sending your data in on time. You're not sending your data in time. And that's because we're monitoring accrual, we're monitoring adverse events, there's interim analysis, there's the Data and Safety Monitoring Committee. Every group meeting, there's a meeting of the SWOG Data and Safety Monitoring Committee looking at existing data on ongoing studies that you're submitting data to. So we need to get good quality data all the time because decisions are being made with respect to should we modify the protocol, should we stop accrual, should all sorts of things. And that there's a slide basically talking about the SWOG Data Monitoring Committee and how it needs data to make those critical decisions. I'm looking at the time here. But, you know, the one thing that's absolutely critical on our side is we should not be asking for stuff that's being submitted that isn't critical for understanding. So every element must count because everybody has to feel like, every patient needs to feel like that if they're submitting something, it has to be appropriate to learn more about what's going in the study. So this is a bit of a story. Lung MAP is our big platform study in lung. There was a positive phase 2 study recently reported, S1800A. It showed a difference in overall survival for a new therapy versus control. Again, you can see the blue arm is the new therapy. The standard is the red, and there was a difference, the vertical difference. You could see thinking of one other way of looking at these curves is looking at median survival. People sometimes talk about median survival, so you kind of draw a horizontal. You can draw a horizontal line across here, and if median survival, you go across 50%. The median is that for the blue arm, and that for the red arm. So it's 14.5 months for the new treatment versus 11.6, which in this group of patients this is survival data. It's a big deal. So the outcome of that was actually a new study, which you may be aware of, called a randomized phase 3 study looking at a very similar randomization to get the formal data for a real FDA conclusion here. And one of the things we did was strip down a lot of the data collection on this. We as part of it was totally minimal data collection. So in other words, these forms are far less than we see in another SWOG or another NCTN group study. And our hope was to get, because in this setting, we thought like our big conclusion would just rest on this bigger group of patients, could we see a difference in overall survival? We didn't need to ask for AE start and stop dates and attributions on every level of every grade of every toxicity and have so much stuff. So so it's a two-way, all of this is a two-way street. We can't ask for anything more than we need, but what we need to make things really draw strong conclusions. And sure. Notice of death. So high quality, timely data are essential for our goods, all studies, and really the team effort is needed to get high quality data. It's just hard to convey how much when you're analyzing the data, how important all of these conclusions right on these things. You always hear these stories of some result in some newspaper for something that that's supposedly to work better. And at the back, you have to think to yourself, who are people who are actually critical to make that result happen? And it's really the data, how the data are coming in from the institutions, from you all. So do you have any questions? I think I went over by a minute. I really did stand in the way of lunch. But I just want to thank you so much for coming. It's also just great to be back in person for some of these meetings as well. And if you have any questions, please track me down here or today, right now, or if you're out having lunch, grab me again.